Good morning, and welcome to today's meeting entitled Unlocking Complex Cell-Based Gene Therapies. Today's workshop is co-hosted by Friends of Cancer Research and the Parker Institute for Cancer Immunotherapy, and we very much thank them for their partnership. I want to thank all of you for joining us today, and welcome to all of our virtual attendees as well. In total, while it feels intimate in the room here, we have over 1,000 individuals that are either in the room or online today, and we appreciate your interest in this important topic. Development of cell and gene therapies represents a rapidly evolving science that holds the potential to become a new treatment paradigm for many diseases, including cancer. To date, 7 cell-based gene therapies have been approved. While applying lessons learned from the recent clinical development programs will most certainly inform the next generation of cell therapies, new approaches may also be necessary to think about as we think about ways to maximize progress. Exploring new models for discovery will be a theme of today's discussion. This, of course, requires careful consideration as the field aims to identify the most beneficial gene modifications and capitalize on novel technologies that are available today. I'm truly excited about the potential of the work and the implications of today's conversations and what they will do to the future of medicine. I especially want to thank Ute Dugan and her whole team at Pisces. Given the large number of virtual attendees today, we're going to do our best to strictly follow the times on today's agenda, and we appreciate everyone's cooperation from the panels and the audience to help us stay on time today. Following each of the panels, there will be an opportunity for audience questions, so please utilize the microphones that you'll find in the aisleways after each session concludes their initial discussion. We simply ask that you briefly identify yourself and preferably keep your questions in the form of a question and on topic. For those of you that are joining virtually, please use the chat function that you can find on your Zoom application in order to submit questions for the panelists. We'll have members of the Friends and Pisces team that will be monitoring the chat for each of the session moderators, and we'll get as many audience questions as we can for each session. So now I'd like to start things off today and introduce our first session. I'll invite our panelists to come up. It will be moderated by former Washington Post reporter Lori McGinley and feature 2 remarkable leaders at the FDA, Principal Deputy Commissioner Dr. Namandjé Bumpus, and Director of the Center for Biologics Evaluation and Research, or CBER, Dr. Peter Marks. Thanks so much for joining us today. We look forward to the discussion.
Jeff, thank you so much. I appreciate the introduction. I am so thrilled to be here. I see a lot of familiar faces. It's really fun for me. And to have a chance to talk to such distinguished guests about this important topic. So let's get right to it. Dr. Marks, you obviously have been instrumental in the FDA's approval of many cell-based gene therapies. Tell us a little bit about how the volume has changed over the last couple of years and what you're anticipating over the next couple of years. What's on the horizon?
Yeah, so thanks very much. Thanks everyone for coming today. So we're in a place now where there is a subtle change. I think we started out with, in terms of chimeric antigen receptor T cells, an incredible, it was like one of those kids' soccer pileups. Everyone was making CD19 CAR T cells. And they were all in the autologous space with a couple of outliers in the allogeneic space. Now we're starting to see branching out of this space where much more complicated, even in the CD19 space, much more complicated constructs moving into the allogeneic space from autologous and branching out into other areas where I think moving from the kind of core hematologic malignancies where we started out in acute lymphoid leukemia and non-Hodgkin's lymphoma, moving into multiple myeloma, but now starting to see the movement into hopefully making a dent into solid tumors, made possible in part by kind of the revolution in genome editing that came with the introduction of CRISPR. So I think a lot of activity here, and if you look at the kind of the pie charts, you kind of see the activity is increasing, but the greatest increase is starting to see other constructs besides the CD19 CAR T cells taking on an important role.
Dr. Bumpus, with all this complexity and the branching out into different things, how is the FDA scaling up its capacity to handle this rapidly evolving field? What are you doing?
I think you just heard Peter and his team have a really big job ahead, certainly with the range of therapies as he was outlining. So I know that he's scaling up, and he can probably speak to that. But it really requires a range of expertise. It's the science, the manufacturing we know is a key component, and people understand that. I know we have a couple of our bioethicists here, and we need to think about all of those things. My job as principal deputy, I really feel, is to support the center directors and the programs and the work they're doing. So Peter's the one working with his team to do the scaling up, but I really try to ensure that I can reduce or minimize any barriers they might have to doing that. So it's really a partnership in that, but certainly being led by CBER.
Yeah, I mean, I think the point here is, as we see the need, I think we will potentially have to hire up and continue to hire up in this area. I think we've made really good progress. You'll hear from Nicole Verdun later, but under her leadership, the office has done a really good job hiring up the PDUFA headcount, the prescription drug user fee headcount that was given to us as part of the most recent user fee negotiations. And there is part of that is a capacity adjustment based on the number of applications that come in, and so hopefully we will continue to staff up as need be. Additionally, we'll staff up hopefully to meet some of our pilot programs if they show that they're successful in meeting the needs.
So are you confident you'll get the staff you need with the appropriate level of expertise in this area?
Well, it's always a challenge, right? It's always because you want the best possible staff that we possibly can, and I think we've done a pretty good job in doing so. Some of that is a little bit dependent on what the environment is. I mean, I'd be kind of Pollyannish if I didn't say that, it changes over time. Right now, it's not a bad time because I think there are a fair number of people that are skilled in CMC, chemistry manufacturing controls, that are out there that we've been able to hire. Physicians that are earlier on in their career who we've been able to hire. So I'm pretty encouraged with the people who we've brought on board recently, and I think it's exciting because we have a diversity of experience and expertise that will serve us well for these products.
And you mentioned the pilots that are really important, I think, with trying to continue to facilitate that innovation and that ability to move these things forward. And like you said, that will give you a real idea about where you need to scale up and where things are coming. But the pilots, I think, are really exciting.
Let's talk a little bit about manufacturing, which I know is one of Dr. Marks's favorite topics. It's incredibly complicated in this area, and you've talked about the challenges and the bottlenecks that present themselves. What can the FDA do about that? What is the FDA's role in that? And do you see a way to do decentralized manufacturing? Would that ease the bottlenecks? And what are your thoughts on that, and whether the current regulatory regime could handle that?
Yeah, so what we're dealing with for complex biologic products is a challenge, right? Because you have the products that we're dealing with here are cellular therapies, which cells on their own want to do what they want to do. And trying to make them reproducibly so that they become what we're used to as a biologic product, which is something that is relatively standardized, is a challenge. And so this is part of what we have to deal with in this cellular manufacturing space. It's also to make that happen because of the clean rooms and the fact that many of these processes have been done to date by hand. It's an expensive process. So I think what we're seeing right now is the emergence of kind of different tracks. So one track is that we'll probably talk about sometime later, but maybe not so much right now, but this idea that you have allogeneic products that work as well as some of the autologous products. We can talk about that. The idea there is centralized manufacturing works there. As a way to potentially develop efficiencies. But let's concentrate on autologous products. So there we're starting to see kind of a wave of automation. There are now more than one company in this wave of essentially practically fully automated production centers where basically you load the vector that you want to transfect and you load the cell product, and in 7 or so days pops out a cell product, or less or a little bit more depending on the various parameters. So automation may really help here. There are obviously, the benefit of that automation is that whether it's done on a smaller device or a larger device, where it's semi-automated or fully automated, you can have a decentralized model, which does bring the advantage of potentially bringing the product closer to home where a patient is, potentially allowing for more innovation in product development. The downside is if you're making a product that's going to be used in a lot of different places and you want it to be the same, you have to make sure that if you want to make the same product in a decentralized manner, that you have the right quality controls in place, the right ways of making sure you're making the same thing every time. And that can be a challenge, but it's addressable. So there are those who are ardent centralized manufacturing advocates and those who are ardent decentralized manufacturing advocates, and I think we're here to be kind of neutral in this. I think they're both legitimate ways of going about this. There are advantages potentially to decentralized manufacturing for certain products, particularly ones that are made fewer times. But we're just there to support this process forward, just as long as the ultimate product, whether centralized or decentralized in manufacture, is of the same high quality.
Dr. Bumpus, I want to circle back to what you referenced, which was the pilot projects. You have talked about the importance of the FDA being nimble and agile in trying to deal with these rapidly evolving technologies. How do the pilot projects fit into that?
Well, I think one thing is the communication. CBER having really open communication with the community, and when I've gone out and either talked about it or heard Peter talk about it, there's definitely a lot of enthusiasm for that, because I think that you really can learn and streamline by having that partnership and that open communication. So I think that's something that's key. But what also comes to mind, especially with the comments we're just hearing, is the depths of scientific expertise around this that exists within CBER. So as you mentioned, we might be neutral or agnostic to approach, but definitely have to have understanding to have those conversations. And CBER has a very robust scientific community that understands a lot of this, not only through the conversation, but even through their own scholarship and research trying to advance these areas. So I think that's something that people sometimes don't remember about FDA, is that we are a scientific agency. We do have scientists who are working to advance things in a way that we can uniquely advance because of this point of view and perspective we have. And I think that our scientists within CBER are a great example of that.
Dr. Marks, you've talked a few times, Dr. Bumpus mentioned communications. You've talked some about lessons, things that you learned from the pandemic and the working on the coronavirus vaccines, and how you might apply that to other things. Are those lessons informing some of your actions in this particular area?
Yeah, so very much so. And although it's not right now directly relevant for cellular therapies, because we're doing it for gene therapies, it's indirectly relevant for one of the pilots. There is a pilot that we're doing, which is essentially a communications pilot. For those of you who might not be aware, for the expediting of the vaccine production process combined really a couple things. It was a matter of manufacturing the vaccines at risk as soon as you had some inkling that they might be effective. But a large amount of what sped their development up came from having constant communication with the people who were developing the vaccine. So whether they were slightly smaller companies or very, very large companies, we had a process during that vaccine development process where nobody stood on the ceremony of our normal type ABC meetings, which take from 30 to 75 days to schedule. What we had was a project manager who the company contacted with a question of, well, how many conformance lots are you going to require for an emergency use authorization? And rather than wait, oh, prepare a briefing book and we'll have a discussion 30 days from now, we actually just said, okay, let's get on the phone tomorrow morning and get that sorted out. That kind of communication happened many, many times, and you could imagine if you get rid of a lot of 30-day or 75-day waits, development can occur much more rapidly. So we hypothesized that for other products, that kind of communication could help, particularly ones for serious diseases, cancers in particular, serious rare diseases. That we're doing a pilot right now, it's called the START pilot, where both the Center for Biologics and the Center for Drugs are looking at 3 products. Ours are in pediatric rare diseases. CBER is in neurologic disease. The idea here is to say, okay, if we give that same kind of constant communication with the sponsor, do we accelerate this measurably in terms of reduction of time? Because even a 10 or 20% reduction in time of development can really have an impact on patient lives when you're dealing with a life-threatening illness. So if this pilot indicates, and it's measurable, there are multiple measurable things in terms of time to development that we can measure and we can get feedback from sponsors on, that if this is successful, then we may be going out and hiring more people because it's a very labor-intensive process. But if it ultimately cuts back the time to development, any MBA in the audience can do a net present value calculation and show that it doesn't take that many products that you shorten the development time by 10 or 20% to more than make up for hiring another 50 or 100 reviewers in terms of the cost.
Well, and tell us a little bit about the other pilot project, which involves working with international regulatory bodies. I'm very interested in what both of you think about how they can move things along for rare diseases and other diseases. Can you give us an update on that?
Yeah, I can start us off, which is that, you know, this is the second thing coming out of the pandemic, which was during the pandemic, we became closer with our regulatory colleagues, both in Europe, Japan, in Canada to the north, and even to the south in Brazil. We got to know each other more and realized that there are differences in our regulatory requirements, some of which are more barriers to entry than actually logical in their nature. And so the concept came up of, could we start to try to break down some of those barriers? And could we do also another thing at the same time, which is, as we try to break down barriers and come to some kind of regulatory convergence, could we also at the same time help educate some of the low and middle income countries about these products so that potentially in the future their populations, they will be ready when their populations might benefit. So there are essentially 2 parallel efforts going on. The first is an effort to see whether we can come to some better regulatory convergence and even collaborative review with our European partners and potentially partners to the north and east. That is called the CoGen pilot. That is currently getting underway. We're first starting with European Medicines Agency. It's very much, for those of you who are familiar with Rick Pazder's Project Orbis, it's taking that as a base, but trying to expand it both to a convergence effort and to a review effort that involves EMA, which Project Orbis doesn't. So our first piece is just making sure we can align with the European Medicines Agency, which has enough of a different structure in how they review products that we have to make sure we can get alignment. And then once that gets working, we'll expand it and hopefully have more regulatory convergence. At the same time, we're working with the World Health Organization in an effort that started before the pandemic. And in fact, it was my last trip to Geneva before the pandemic, to try to develop a global regulatory framework for these products, cellular and gene therapies, so that it makes it easier both to have alignment in high-income countries, but importantly, to start to educate low- and middle-income countries about these products so that when they make it into those regulatory environments, it's easier for them to be accepted both by regulators and potentially by populations. And that actually is moving along nicely. A white paper was published, and actually next week there's a meeting of global regulators in Oman that I'll be chairing that is trying to move that forward. So we'll see where that can get us.
I was going to just add, that international collaboration, I think, is a really key component to encouraging that innovation. I think if, folks see that there's that alignment, then it does make maybe, more willingness to put forward some of these more innovative and novel ideas about potential therapies. And, as was mentioned, it's the idea of even doing potentially some joint reviews or even thinking more broadly at the agency about how we can coordinate internationally on, say, certain scientific methods and platforms that might be part of various regulatory processes. So even the regulatory science aspect. Recently we had a bilateral FDA-EU bilateral meeting on campus at FDA, and this was a lot of the topic of discussion. So I think Rick Pastor and Peter certainly leading the way there, and a lot of opportunity even around kind of the science components of things, I think are real opportunities, and that this will help us get an example to help move that forward more further.
I just want to stop for 1 second to say that we are going to be taking questions in a few minutes, so there are going to be microphones there and there, I believe. So in a few minutes, if you would like to make your way to the microphones, then we'll be taking some questions from the audience. Dr. Bumpus, looking forward, do you see other, we've touched on several issues now that affect these cell-based gene therapies. Do you see others that the FDA needs to be focusing on to make sure that it's ready to deal with the next generation of treatments?
I think that there are certainly, we'll see more and more applications of these approaches. I think, autoimmunity and thinking about, how you move this forward and develop it without impacting kind of protective immunity. So I think there's a lot of work to do there. I think increasingly we'll see AI involvement in understanding how to do some of this. So I think there's a lot that we can do. From my post, as I mentioned, I'm really interested in not only how do we enable the centers to do this, but how do we advance our science internally so that we are ready as those questions come and that we can identify gaps and even do the research that we need to do there as needed.
And there are opportunities to use these therapies for other diseases, are there not? What are some of the things that they might be used for?
Certainly. So on my mind, like I said, things, neurodegenerative, autoimmunity, what are some of the areas that you're thinking about?
I mean, you're talking about for the cellular therapies, I think the chief among them are autoimmunity, some infectious diseases applications even, and a whole wide range of both hematologic and solid tumors. So it's a pretty broad scope of where we might go with this, given that there are a lot of autoimmune diseases out there.
Dr. Marks, let me ask you something that's a little more specific about this meeting. As you know, many of the people here are interested in these therapies and how they might be able to be made to work more effectively for solid tumor. And there's going to be much more on this topic over the next couple of hours. Can you just give us your preliminary thoughts on testing various T cells with different gene edits in patients?
Yeah, so I think there's a desire here to try to take what we know, and I mean what's happening right now, and whether it be because of CRISPR becoming an incredible tool that allows us to have an incredible power over editing the genome in multiple genes, or producing multiple cell types, basically cells from someone, each with a different gene altered, or each with multiple different genes altered. I think there is this desire to see, can we, as fast as possible, come to some improvement in the current state of efficacy. I think Dr. June's work has indicated that certain, if you have certain changes, you can get a better CAR T cell. And the question is, can we figure out a way as fast as possible to essentially make certain genetic alterations so you have essentially the fittest of the CAR T cells? In other words, it's almost like it's almost expedited evolution. And for us, it's a matter of balancing, how many changes you can make with ensuring that we feel safe putting those changes into a human. And so I think a lot of the discussion you'll hear today is kind of this balance of we want to help facilitate innovation, help facilitate progress, but need to balance that with just making sure that we're comfortable with the number of changes or the number of different types of cells that go into a person so that we are comfortable that there won't be something untoward. And it might be that this is something that evolves with time. It might be that we put one of the things that FDA does do well is we sometimes put our toe in the water. That's the way I get into the pool. It's very slowly. And we kind of get in slowly, and then eventually, we're in the deep end.
Thank you very much. Are there questions from the audience? Well, in that case, thank you. Oh, I'm sorry, couldn't see you.
Hi, I'm Maisha from Inside Health Policy. So I have a question because AI algorithms are known to evolve as they absorb new information. But to protect the integrity of a clinical trial for an AI-based cancer vaccine, Moderna had to lock it down to avoid any changes that may have co-founded the results. So how much change to AI algorithm is allowed before the vaccine should be considered completely a new product?
I don't think I can speak to a specific question like that, but say that, I think it's the issue of, I'm going to use it to say that, look, we're happy to have discussions of what AI can and can't do, just so that everyone knows AI is not writing reviews for you yet at FDA, nor though you might think it, our reviewers actually AI bots. Those are actually real people. It obviously is something, all kidding aside, that we have an AI committee that's really trying to think about how you actually do think about this and how we can potentially incorporate this. That question may be a little bit of a step ahead of some of where we are right now.
And we have some research in, say, the AI space preclinically to think about how you use AI to make preclinical safety predictions and things like that, just right now at a very basic science level of things, but certainly a lot of interest in how we can help advance application of AI in regulatory science.
Well, thank you so much, and I'm going to turn the proceedings over now to Dr. Rebus, who is going to handle the next section. Thank you.
Well, we're on to a great start of this workshop. I want to thank Dr. Bumpus and Dr. Marks for the very insightful and interesting and informative discussion, and Laurie McGinley for your outstanding moderation. We're going to go to session one, and session one will have 2 academic presentations and then a panel discussion. So they're putting the chairs here for the extended panel. And I'm so pleased to present the first speaker, Dr. Carl June. Carl is professor of immunology at the University of Pennsylvania. He's a pioneer in CAR T-cell therapies. You've also already heard him being referred to. He's done seminal contributions to many parts of the field of immunology for cancer. He recently received the Breakthrough Prize, and I'm not going to list all of the other prizes that he's received because we wouldn't have much time, so I want to thank Carl for being here.
Thank you very much, Tony. And it's really an honor to speak here on behalf of really the Parker Institute, where we've come together to help present some consensus in the scientific areas. And looking for my slides there. And I'm going to address chimeric antigen receptors within the space of cell and gene therapies. It's just one example of many possibilities. And my disclosures are here, which I'm going to present some material that is conflicted with technology license to Novartis from my laboratory. And this is an outline of what I'd like to show, some status of where CAR T cells are today, where they began, and then one issue that's come up recently, which is this issue of secondary cancers in CAR patients, and then the most recent, which we just heard some about, CARs with autoimmunity. So, for a long time, cancer was thought to be just a disorder of the genome, and shown here from this famous Doug Hanahan figure, but recently the field has concluded that a cancer, to be a cancer, not just needs oncogenes and so on, but it needs to avoid immune destruction. And that's led to how maybe cell therapies could be so potent to try to overcome that and eliminate cancers. And for many years, we've had the ideas of immune therapy for cancer, and it just unfortunately never worked for the complexity of what we didn't understand at that time. But since the 90s, there's been a lot of progress both in vaccines and cell therapies. And in 2012 then, or 2013, science said, cancer immunology with CAR T cells and checkpoint therapy was the #1 breakthrough of that year. And there's been a lot of momentum since then. So with Stan Riddell and Ton Schumacher, we wrote a paper about a decade ago on the major types of cell therapy. There are both cell therapies derived from the tumor. Those are tumor-infiltrating lymphocytes, which just this year became FDA approved. And then there are T-cell receptor-modified cells and CAR-modified cells, and I'll be addressing CAR-modified cells today. This is a radar plot of the therapies that are FDA approved and marketed in the center where the bullseye is. I like this plot because it shows in red allogeneic products and in green autologous products. There are both peripheral blood T cells, NK CAR cells, for instance, CAR macrophages, et cetera, many kinds of cell types being developed, and both autologous and allogeneic. So today we're focusing on autologous cell products. I want to emphasize that. I think there are different issues with allogeneic cell products, but they're going to be equally important, I think. So with Michelle Sadelain, a few years ago, we wrote a review on the status of cell therapies with TCR-modified cells and CAR-modified cells. The main advantage of a CAR cell is that it's antibody-directed. It does not have to be matched to the patient's HLA type, unlike the T-cell receptor, but both of these are very potent and will, I'm sure, be developed and become central. So when that was published, or the first FDA approval of the CARs in 2017, there was a lot we didn't know. I mean, that we had data that they were very effective in pediatric and young adult leukemia. When we first started CAR cells, the first approved ones have a mouse antibody for, to bind to CD19, and we thought they would be rejected because that's always happened. And it turns out we've had CAR T cells now, even though they have a mouse product, with CD19 can persist for a decade in patients as a living drug. We're also worried about this issue of transformation, so-called Frankenstein cells. Would genetic modification lead to transformation of T cells? That's a very rare event with autologous cells. It's, unfortunately has happened initially with early stage vectors with gene-modified stem cells. And there, the issues of transformation are more serious than with autologous T cells. And we still don't know the real profile and risk with allogeneic cells, although it looks like allogeneic cells may have an easier way to transform if their T cell receptor is not working. So we began CAR T cells here, just a handful of academic centers in the US about 20 years ago. And now, when I did a search last night in clinicaltrials.gov, there were 1,100 CAR T cell trials listed, and very surprisingly now, 44 listed for autoimmune disease, which is a very recent evolution in this space. Today, we're focusing on the cancer use of CAR cells. So CAR cells to become a reality required three major innovations. One was the retargeting of the T cell to be antibody-directed, not MHC-directed, as I mentioned. The second was we had to devise ways to grow efficiently autologous T cells, since there are no universal donors for T cells, unlike for blood, transfusion medicine. So for autologous cells, there's an ex vivo expansion step. And then the third aspect that needed to be developed was methods to efficiently gene modify T cells. And that was done initially at the NIH with gamma retroviruses, and then at the Dana-Farber by Michel Sadelain, where he actually used those to modify T cells with gamma retroviruses. And then we conducted the first trial in humans with gene-modified cells using HIV-based lentiviral vectors. And those are what are now comprised in the commercially approved both gamma retroviruses and lentiviruses. And the actual first CAR trial was done in HIV, and this was during the AIDS epidemic when there were no medicines, and with a company called Cell Genesis, we did a trial with a CD4 Zeta CAR back, a series of 3 or 4 trials in the 90s. And this is a CAR that didn't have any co-stimulatory aspect to it. It just had the Zeta chain of the T cell receptor, and it can eliminate HIV-infected cells by virtue of the CD4 extracellular domain binding to GP120 of HIV that would be found on a CD8 infected cell. And we had some efficacy and we had a lot of safety in that trial. But when the drugs came along, that approach for antiviral and HIV, the cell therapy was abandoned pretty much. But because of the FDA's really forward-thinking process and of having longitudinal safety follow-up on patients who are recipients of gene-modified cells, we had about 40 samples that were frozen from the patients treated on our 3 trials, and we looked to see if they were still engrafted. And we found that the patients still had these CD4 Zeta cells in them a decade after infusion, and that the half-life of decay of those by 3 different statistical methods was about 17 years. So it was really a first demonstration that you could have with autologous T cells, a living drug, after gene modification. We're now following some of those patients out to 20 years, and I can say in unpublished results, they still have CARs, and there has not been untoward safety events. So we have this decade-long safety with gamma retroviruses in 40 patients with AIDS now. And then 2 years ago, we published in Nature the first follow-ups of our initial patients treated with lentiviral modified cells that were targeting CD19, so CD19 CAR cells. And we found that those cells have now survived actually more than a decade in the initial patients treated. So CAR T cells can be living drugs, which is why we've been attracted to them, the fact that they can potentially be a lifelong, both to eradicate a tumor and then stay active for immunosurveillance. So our first patient treated with what became Kymriah was in the summer of 2010, and we then treated many different pediatric and adult malignancies with what is actually the same drug, which is an autologous gene-modified cell with a CD19 CAR, and it's been approved on labeled basis for different forms of B-cell tumors. And our first patients treated with CLL in 2010 remained in remission for a decade or more now without any evidence of recurrent leukemia. And then our first pediatric patient was treated in 2012. Her father is here. You'll see her later. And she's actually a freshman at the University of Pennsylvania, I'm happy to say. So, these patients have had long-term follow-up and safety. We now know that we can dictate how long a cell persists in patients based on the signaling domain. So we can have more rapid proliferation, but shorter persistence with the CD28 signaling domain, and 4-1BB signaling domain gives longer proliferation and slower expansion, but longer persistence in patients. And we found out that these CAR memory cells in patients, at least when it's a CD19 target, maintain slow tick-over proliferation in the bone marrow. We found now that by reports from companies, more than 34,000 patients have been treated with for leukemias and lymphomas and myelomas. Allogeneic CAR products are in earlier stage trials, and the safety profile of allogeneic cells is less mature than with the autologous cell products. And the real issues, as Dr. Marks mentioned earlier, is really automation and cost of goods for autologous manufacturing, and there's been a lot of progress in that area. So one thing we have learned by following these patients is that not all T cells are created equal. So you can insert the CAR, but it depends on the CAR signaling domain and then issues of where the lentiviral vector or retrovirus inserts. And you can get gain of function or loss of function depending on where these insertions are. And we found in the 10th patient we ever treated a clonal outgrowth of cells that had the insertion of the lentivirus into the TET2 gene, and reported that in Nature in 2018. It gave the cells an advantage over the cells that didn't have the TET2 loss of function. And then from the NCI, they reported the clonal expansion of CD22 CAR cells that it integrated into the CBER gene. And again, that gave improved performance of the CAR cells over the cells that didn't have that happen. And that's been an impetus for why we've come together today to see how we can do this in an intentional way. And so we looked at this in 2020 with the patients we had, and this is work done by Rick Bushman at the University of Pennsylvania, looking both at the infused product, which has on average about 1,000 different integrations from the lentivirus in different areas, and looked at the genes where they were inserted. And this was when we had 40 patients treated that were analyzed in depth, both at the time of what was infused and then following up in the patients over time. And we found at that time 6 out of the 40 patients had integrations into the TET2 gene, and that gave an advantage to those cells. And we've now done and found, with about 1,000 integrations per patient, and with the amount of patients treated with usually an infection, multiplicity infection, 1 or 2 vector copies per cell, that we've really done saturation mutagenesis of the entire human genome now with the amount of patients treated and in the infused cells. And so we're now looking at this in more depth, and this is unpublished work that's being peer-reviewed at this point, but now with about 50 patients with ALL and CLL, we found about half of the patients have integrations into TET2. So this is actually 113 patients, and 36% of the patients have TET2 integrations if they had CLL, and 51% have ALL. And on the right side of this, on top is the CLL patients, and the bottom the ALL patients. And the TET2 gene, which is on chromosome 4, these integrations are tiled throughout the gene, the transcriptional unit. Lentiviruses, usually integrate into active transcription units. And so there's no hotspot here, but it's throughout the gene. And what we find is that gives the cells an advantage over the wild-type cells that don't have the TET2 integration. And this is further work now led by Joe Frieda's lab at the University of Pennsylvania, where he's used an AAV6 system to knock 2 genes at once into T cells. One is site-specifically into the TRAC gene to put a CAR in there, a CD19 CAR, and the other one is into the TET2 gene, where he put the EGFR gene, so it could be a suicide system. And in the center then is the manufacturing and then repeated stimulation with cancer cells that have CD19, and on the right you can see there's about a 10-fold growth expansion of the CAR cells if they also have an integration into the TET2 gene, which causes loss of function. So it's a target-dependent gain of function by intentional knockout here of the TET2 gene. And so that phenocopy is what we found in the patients. And then to further look at this, Joe's looked at in an NSG mouse model the expansion and anti-tumor effects of CAR cells that have the TET2 knockout and the CAR knocked into the TRAC locus, or just the CAR into the TRAC locus targeting CD19. And that's the group of animals on the right, bioluminescence tumor burden. They work much better with this intentional knockout of TET2. So that's one of our favorite genes to knock out, but there are hundreds that have come up from the work that you'll see, for instance, from Alex Marson and Marcela after I'm done. So what we found, initially by discovering from a patient, reverse bedside back to bench, was that, we had clonal outgrowth of CD8 CAR cells in patients with CLL, and they work better. And we've now recapitulated that intentionally in patients in the TET2, both in mice and in human T cells. And so it's really a powerful way to be able to study the T cells in patients after treatment because they're genetically marked and we can see how they evolve following infusion. So then, this comes in naturally with genome editing because we have many tools such as zinc finger nucleases, TALENs, CRISPR-Cas9, base editing, where we can literally rewrite the human genome now. It's really, I think it's fair to say when we started CAR T cells, none of us thought this would be possible. And so this is part of why we're here to discuss this today. The first human genome editing trial we actually did in HIV patients in collaboration with Sangamo Biosciences. And in that trial, we knocked out the HIV co-receptor gene, CCR5, in order to phenocopy patients who have the Delta 32 mutation that gives you loss of function on CCR5 and resistant to HIV. We found out that we could do that in that trial and that it was safe and that the cells had long-term engraftment after intentional knockout of CCR5, and they had a competitive advantage in the patients over those cells that had wild-type CCR5. And then a few years after that, with the advent of CRISPR-Cas9, we now could do multiplex editing, which is difficult to do with zinc finger nucleases. So we used, in this trial, 3 different guide RNAs on autologous T cells to knock out the T cell receptor alpha gene track, the beta chain, and then the PD-1 gene in patients with refractory cancer. And this was a pilot study done with the Parker Institute. And 4 patients were manufactured in that trial. So on the upper left, it shows the manufacturing scheme, which was to introduce ribonuclear protein complexes with 3 guide RNAs to knock out the 3 target genes, which are on 3 different chromosomes, and then use a lentiviral vector to introduce an HLA restricted T cell receptor to the New York ESO cancer testis antigen. And we found that those cells had the same expansion, and we harvested after 12 days, as shown on the upper right. And when we did single-cell analysis of the infusion product, one patient is shown on the lower left, with the technology from this back in 2017 with CRISPR-Cas9, we had about a 50% knockout efficiency, and we found some cells had single knockouts, for instance, of TRAC 11 out of 27 cells, but some cells had all 3 knockouts plus the inserted transgene, 6 in this case. And so we found very rare off-target effects. We had deep analysis on this. The guide RNAs had high fidelity and found very little evidence of off-target editing. What we did find was a 1 to 2% incidence of chromosomal translocations. As I mentioned, each of these target genes were on different chromosomes, and Cas9 as a nuclease, makes double-stranded breaks, and sometimes in the reannealing creates chromosomal translocations. What I didn't know until I started getting deep into this is that we all have chromosomal translocations. None of us have perfect genomes. So when you look, and there's literature here, shows that with sensitive assays, your B and T cells have translocations. This occurs during recombinase making of the T and B cell receptors. So it's a natural part of B and T cell life is that chromosomal translocations do happen. Even the Philadelphia chromosome is in healthy patients when you look with sensitive means. So that was the main complication we found. What we did find in that study was that those cells had decreased fitness if they had a translocation, compared to the cells that didn't. So by following the infused cells in patients where there was initially a 1 to 2% frequency of chromosomal translocations, they went down in the patients over the next 3 or 4 months. And so that was the result from that first multiplex genome editing study. So In summary, where the field is now, publications with feasibility and safety using zinc finger nucleases, TALENs, CRISPR-Cas9, and base editing have all been done with human T cells, and it gives us really a possibility to screen through hundreds of genes now to identify the combination that may give improved performance in solid cancers. Now, we all got a cold, slap when last November we learned that there are secondary malignancies in some patients after CAR T-cell treatments, and this is an ongoing issue that the field is working at with the FDA. So the summary back in November was that there were 22 cases reported to the FDA's database for adverse event reporting system. About 3 cases have had where the lymphoma is CAR positive, and there's now a new black box warning for the commercially approved CAR T cells on this. So this is an ongoing issue, and we have some data that's unpublished that I'll show from both Penn and Stanford. So the transformation of genetically modified autologous T cells is a very rare event. It's when you intentionally introduce oncogenes into human T cells, they do not transform. They're resistant compared to fibroblast or stem cells. And then that's been reflected in the natural history of following our patients given autologous gene-modified CAR cells when done as specified by the FDA with multiplicity infection, 1 or 2 gene copies of the transgene per cell. But mature T cell transformation can happen under unusual settings. There's a reference there. 2 down there, one is a report in Blood where allogeneic CAR cells using a Sleeping Beauty transposase system, 2 out of 9 patients got CAR positive lymphomas within several months after infusion. And so now at Penn, we've gone back and looked in detail at 783 patients treated on our trials, beginning with HIV CAR patients 20 years ago and up to where we currently are. And we've used in these studies gamma retroviruses or lentiviruses. We found secondary malignancies in 18 of the patients, 2.3% of the patients, with a median onset shown there of about 2 years. And solid tumors were the most frequently identified secondary tumors. And we found in none of the cases where there insertion of the CAR molecule into a secondary malignancy. So nothing where we could directly attribute the CAR to a secondary cancer that occurred in the patient after treatment with gene-modified T cells. And this is work, this is what we found in studying these patients, the 700 and something patients. What are the top most frequently integrated genes? And again, we found TET2 to be the most common, and the list of most common insertions is shown here. So there's a number of genes that are favored in patients when you have long-term follow-up, and these are things we want to test in patients now to do this intentionally to see, because as I mentioned, none of these patients have had a secondary CAR-positive lymphoma. And this is work now with an equivalent amount of patients at Stanford from Ash Shalidazad's group. 725 patients were treated, and they found secondary malignancies with the most, 11 were solid tumors, 13, which the most common was myelodysplastic syndrome or AML, and they had only 1 case of a T-cell lymphoma, but that was an EBV-driven lymphoma, and it did not have integration of the CAR transgene. And this is the model that they've come up with in Stanford, how this can happen, where there may be clonal lymphoid poiesis at the beginning of what the patient has, and then they came down with a B-cell lymphoma, and they have a baseline amount of mutations there, and then they also have come down later with this, from the B-cell lymphoma can get a T-cell lymphoma based on their underlying clonal hematopoiesis. So it's a rare event, and it does occur in patients. With this between Stanford and Penn and these unpublished studies with 1,500 patients analyzed, there's not a single case where the integrated vector transgene has been observed. We think that, we need caution if patients have extensive mutations before therapy, and then also when applying CAR therapy to non-malignant conditions, such as autoimmunity. And then there's been shown that there's this, by a study published by Chihara et al., that there's a bidirectional increased risk of B-cell lymphoma and T-cell lymphoma. So if you have a B-cell lymphoma, you have a 5-fold risk of, even without being treated with a CAR, of getting a secondary T-cell lymphoma, and then reverse. And that's basically what the Stanford case just showed. And so the underlying risk of second primary malignancies is also can be looked at from an epidemiologic basis, and this is called a mortal time bias. And that's basically, the old saying, dead men tell no stories. So if you were going to die, we don't know what your risk would be of getting a second cancer later. So if you get a therapy that then prolongs your life, you can then now have this time bias, immortal time bias it's called. And there's a JAMA study about that. So, there's CAR T cells being used in many different indications now on an experimental basis. And then what I've called out now, though, is this really amazing results in autoimmune disease that have come out just this year. And this was led by the group at Erlangen, a series of 15 patients treated with basically a CD19 BB zeta CAR, and they found a 100% response rate in 3 different autoimmune diseases after fludarabine, Cytoxan, and a single CAR infusion. And so this has led to, as I mentioned, 44 trials now listed on clinicaltrials.gov. So 3 different autoimmune diseases were in that work, SLE lupus, inflammatory myositis, and scleroderma. They found that pathogenic antibodies have decreased or become undetectable, and then drug-free remissions induced. So this is a very exciting finding. The safety of this and so on, obviously, is very early days. So to summarize here, CAR T cells, we have all concluded that dysfunction and exhaustion and trafficking are all limiting issues with solid tumors. Synthetic enhancements of cell intrinsic T cell functions are possible, and the risk of secondary primary malignancies and T cell lymphoma after CAR T cell in hematologic malignancies is low but real. Accidental disruption of TET2 and CBER improves CAR T cell functions in patients with leukemia and lymphoma, and hundreds of genes have been identified where disruption may improve function, and screening in humans, I think, will be the best way to identify promising cell candidates with improved function for future trials. And I've had many collaborators at the University of Pennsylvania and Children's Hospital at Stanford in this work, and I'd like to thank you for your attention.
Any question from the audience? What to do? I don't know if we can take online. You told me there were 700 people online.
Okay.
Okay, so, well, thank you.
Yeah.
Yeah, thank you very much, Carl. To have this perspective from when the field started to where we are now and looking at the future, it's just amazing. And to continue to do this, we have the next speaker, Alex Marson. Alex is the director of the Gladstone UCSF Institute of Genomic Immunology and professor of medicine and professor of microbiology and immunology. He's a recognized world leader on CRISPR gene editing. Carl published the first approaches to not only knock out genes, but knock in genes with CRISPR without using viral vectors. And he's pioneered doing genome-wide CRISPR editing in T cells. And that's why we're here. We're trying to see where the science is and how can we better bring it to patients. Alex?
Well, thank you for the introduction, and I really want to thank PICE, I want to thank the Friends of Cancer Research, and I want to thank the FDA, and it's an honor to be here today. It's a particular honor to speak after Carl, who is not only a friend, but really an inspiration for a lot of the work that is done in my lab and by many of us in the field. Let's see. There we go. So what I want to talk about today is really a special moment that we're in medicine and biology, thanks to what Carl alluded to as really just the early days of imagining how we can go in and actually access the genetic code inside of our cells, with a special relevance here for cells in the human immune system. So thanks to DNA editing technologies, CRISPR in particular, now all of a sudden the genome is accessible and modifiable, and we have new opportunities to think about an enormous range of all the genetic modifications that we can make with a very targeted focus on how can we make the cell therapies for cancer and other diseases more potent and safer. And so that's what I'll talk about today, is a set of opportunities that are emerging with this technology. I'll start with trying to advance my slides. These are my disclosures, including some things in the field of cancer immunotherapy, a particular relationship with Arsenal Bio. This is what Carl has really laid the foundation for, and I just want to really emphasize with this moment where we are. Right now, there are FDA-approved medicines, a handful of which are shown here on this slide, where patients can go in, they can have cells from their own blood circulation taken out, genetically modified. That current genetic modification happens, as we heard with Carl, in the FDA-approved versions, either with a lentivirus or with a retrovirus that insert a gene that specify an antigen receptor, that specify this CAR. Other versions, experimental, can be done with T-cell receptors and with an expanding set of different receptors. But fundamentally, what we've done is add a gene that can make the cells recognize something in the cancer cells. These CAR T cells or other forms of adoptive cell therapies get reinfused back into patients, and as we heard from Carl, the results have been miraculous in a huge range of different patients that have now benefited from these. That said, there are still patients, even in the hematological malignancies, that continue to relapse, and where we even have more room for improvement is in solid tumors, where we still haven't really seen the home run that we all want. So our firm hope is that where we are now is really just the tip of the iceberg of what genetically engineered T-cell therapies are capable of. So why do these not work in solid tumors? There's a number of reasons, but I really want to focus on a couple. Even if we can successfully engineer antigen-specific T-cells that can go in and recognize a target on solid tumors, the tumors have a number of ways of fighting back and silencing the T-cells that we've engineered. So even if a T-cell recognizes a target, those cells can become exhausted or dysfunctional as they try to clear out the tumor. In addition, the tumor can secrete things that make it an immunosuppressive microenvironment. Various metabolites and cytokines can be secreted by tumors to try to turn off these T-cells. The cancers can have checkpoints on them that silence the T-cells. And there even can be recruited other immune cells that can be suppressive. So the T-cells somehow have to navigate through all these challenges in a tumor environment. And that means that if we want to design successful T-cell therapies for the future, we have to think about a target profile of what these cells will look like. We want to engineer them not only to have antigen specificity, but to have a number of different features where they resist all these modes of suppression that I just talked about, where they continue to proliferate and accumulate in the tumor, and where they continue to do so in a way where they're effective at clearing out the cancer cells. So how can we do this? Well, we heard from Carl that he started with a reasonable guess about trying to knock out PD-1, a gene that's a checkpoint gene that's been targeted successfully with checkpoint inhibiting antibodies for a number of cancers. And that's a reasonable guess. But I think it's fair to say that we still don't know what the gene modification is that's going to be the ultimate winner for what's going to make the most effective CAR T cells. And so my approach, and a growing number of us, is Let's learn from the biology. Let's let various gene edits compete with each other and see which ones win out. So conceptually, I'm going to show you a video of what we'd like to do. We'd like to be able to take a collection of human T cells and make in each one of these cells a different gene modification. And at the same time that we make those different gene modifications, all the cells could have the same CAR T cell, CAR molecule, or TCR. And then we'd like to race them against each other. We'd like to ideally be able to see in a human which ones not only find the cancer cells, but even as they get into a tumor, continue to accumulate in the tumor and continue to clear these cancer cells. So this is the ideal that we'd like to work through, where we could actually learn and find the winners inside a human tumor. So how close are we to that, and what steps need to be done? So this is the existing clinical manufacturing pipeline. What I'm saying fundamentally is we'd like to add a step. We'd like to take advantage of the power of CRISPR to take an existing manufacturing where there's either a fixed CAR or a fixed TCR, that stays the same, but then we add a second step where we introduce gene modifications. Now, what Carl has already done in humans is be able to do one gene modification to combine adding a T cell receptor with knocking out, for example, PD-1 along with the T cell receptor. But we'd like to not limit it to just one guess. We'd like to make many guesses. And so the way that there's technology that now makes this possible, CRISPR is not only efficient at editing sites in the genome, it's incredibly flexible. And we are increasingly expert as a field at being able to direct CRISPR at any site in the genome. And that means that we can actually make a library or a pool of different CRISPR molecules and put a different one into each one of the CAR T cells or TCR T cells. And that means that you could end up with a population where each CAR T cell has a different gene knocked out. And ideally, we'd like to be able to then infuse that into a patient and see which ones continue to grow in blood or ideally even in the tumor microenvironment. And then we could do exactly what I just showed you. We could actually go in and CRISPR, thanks to advances in molecular biology, we have the ability to make these gene edits. We can make them flexibly so that each one happens in a different cell. And with advances in DNA sequencing technologies, we can go in and really read out which are the winners. We can go in and see exactly which gene modifications we've made that are accumulating in the tumor microenvironment. We can even look at the full transcriptome of cells that have accumulated in the patient. So that's what we'd like to do. So how close are we as this to a field? Let me just show you that my lab has been working along with many, many others to build an expanding set of being able to use CRISPR to test every gene in the genome, not in artificial cells, but directly in human T cells. Over the past 5 to 10 years, we've gotten to the place where now we can, in a population of T cells, disrupt or knock out every gene in the genome. There's 20,000 genes. We could get rid of a different one in each cell and then race them against each other. And I won't go into it as much today, but we have complementary technologies where we can start to turn on every gene in the genome in a different cell. We can introduce different mutations into different genes. We can even add different genes to each cell. But fundamentally, what you end up with is a population of cells where each cell has a knowable and measurable genetic edit. And then we can race those cells against each other. And what we've done is do this in a number of different contexts. We can actually put these cells into different stresses that mimic different features of the tumor microenvironment and see which ones will grow out. We can sort the cells and see which ones have features that are predictive of therapeutic success. And we can also couple this with single-cell RNA sequencing technologies to get a global measurement of what we've done to each cell. So let me just give one vignette about one gene that has come out of these studies that we would not have otherwise found. So this is work that was started in my lab by two amazing postdocs, Eric Schifrud and Julia Carnevale. And they developed a system where we could put in a library using lentivirus of guide RNAs that would direct CRISPR to every gene in the genome, but each cell would get a different guide RNA. And then we would add the Cas9, the CRISPR nuclease, to all the cells, and you'd end up with a population of cells, human T cells, where each cell has different genetic edit. We could then separate the cells when we re-stimulated and see which ones divide more or divide less as a result of the genetic edit that we've put in. And basically, as we separate the cells, we can just count up which genetic edits occur more frequently in the dividing cells or more frequently in the not dividing cells. And what we could see is we could, in one experiment, learn fundamental pathways of biology. We could find all the genes in one experiment that are required for T cells to proliferate as they get re-stimulated. And that recapitulated a lot of biology that's been known through biochemistry. But we started finding new targets that when you knock them out, when you disrupt those genes, the T cells start proliferating more. We got very interested in these genes, and we started testing them that came out. Many of these had not really been studied ever in T cells before. And we started seeing which ones would make better cancer killers. So we would knock out now one of these genes with CRISPR at a time and then co-culture them with cancer cells that we would label in red. And you could see that we could put them into a TCR cell, and the antigen-specific cells, we could make them better at clearing out these cancer cells in vitro with a number of these genetic edits. Now, we pushed this even farther. We wanted to see, could we learn even more lessons about what would make a better T-cell treatment? And I just told you that one of the things we'd really like to do is be able to make T cells resist the immunosuppressive environment inside of solid tumors. So Eric and Julia went back and they did these tests of every gene in the genome again, but now they didn't just do it under normal conditions. They added to the culture conditions a number of different molecules that we know can be found in different tumor microenvironments to try to experimentally recapitulate these challenges that T cells would face. And across all of these challenges, we could start to see different gene edits that would make T cells resist different modes of immunosuppression. And we got very interested in genes that are all the way on the right side of the screen that would make the cells resist multiple modes of immunosuppression. And at the top of this list was a gene called ROSA2. So ROSA2 had never been studied before in T cells, and if we weren't doing these tests in an unbiased way, we would not have started. This is not where we would have started. So what is ROSA2? ROSA2 has been studied in other cells. It's a negative regulator of RAS signaling, which is important for cell proliferation and metabolism. And if you get rid of ROSA2, our hypothesis was maybe this would make more active RAS signaling. And that's exactly what we saw. We could see that if you get rid of ROSA2 with CRISPR, you could see more active RAS accumulating in the cells. Didn't always accumulate, it only accumulated as the cells were stimulated. You could see more active RAS. Now, for those of you in the audience, that I think RAS will quickly say, are we making these cells? RAS is associated with cancer and malignancy. Are we making these cells into T-cell malignancies? So that is something that is very much on our mind. And that is not what we see. So I'm going to show you 3 lines of evidence that we were able to very stringently test whether or not by getting rid of ROSA2, we're transforming these cells into malignancies. First of all, if we get rid of ROSA2, the cells don't divide out of control. What happens is if you stimulate them with antigen or with other modes of stimulation, they will be more sensitive and they'll proliferate more, but in the absence of stimulation, they're not dividing out of control. In addition, if we withdraw IL-2, which is a cytokine that's important for keeping the homeostatic and fitness or proliferation of T cells, if we withdraw IL-2, the cells die. So they still depend on cytokine. So they depend on stimulation, they depend on cytokine. And if we put these ROSA2 knockout cells, in this case, we did this with a CAR, a CD19 CAR put into the TRAC locus. So this is adding a CAR, removing the TCR locus. If we put these into an immune deficient mice, the mice continue to gain weight normally. Over a relatively long period of watching these mice, we didn't see overt toxicity. Now, that part of that might be that we were getting rid of the T cell receptor in this case, and so getting rid of reducing the risk of autoimmunity. But we didn't see malignancies accumulating in these mice. So what I want to leave you with on this slide is really many of the improvements that we will see will have some features that look like they could overlap with features of malignancy. But T cells, as Carl said, are resistant to malignancy. And even some things that might be oncogenic in some context don't necessarily take us all the way there. But luckily, we have measurable assays like antigen dependence, cytokine dependence, and in vivo toxicity experiments that can give us some measure of comfort that we're not creating uncontrolled malignancies with our edits. Now, the good news is that the results were really astounding. Not only was it safe, but if we took out ROSA2, we could make CAR T cells better. So in this case, we're taking CD19 TRAC CARs. This is CD19 put into the TRAC locus, one of the best CARs that's out there, and making them even better. So this is reducing the dose to the point where eventually the control cells with the CD19 alone fail, but the CD19 CAR T cells with ROSA2 knockout continue to control tumors and have improved survival. In addition, with collaborators at St. Jude's, they independently were testing the effect of ROSA2 in a CAR T cell for a model of solid tumors. This is a sarcoma model with an FA2 CAR, and they also saw improvement with ROSA2. So we're really excited about ROSA2 as a really powerful guess. We now have mounting preclinical evidence both of its safety and its potential efficacy. But the key thing is that ROSA2 is not alone. What I want to really leave you with is that that came from our trials testing every gene in the genome. ROSA2 emerged as one of a number of different really promising candidates that's emerging as we do this unbiased genetic studies directly in human T cells. Others are taking complementary approaches. So this is a study from Hongbo Chi at St. Jude's. He tested a number of genes in mouse T cells and tested their effect in vivo to see if you actually infuse mouse T cells with a number of different genetic edits, which one of those cells will preferentially accumulate in a tumor. And he highlighted a gene called regnase that looks promising. If you get rid of regnase, you get an expanded infiltration of T cells into tumor microenvironment. Crystal Mackel, who's here, did beautiful work showing that with a particular CAR that causes tonic signaling, she tested every gene in the genome and zoomed in on a gene called MED12 that continues to look really promising as a way to make better CAR T cells that are resistant to some modes of exhaustion. And then those are unbiased studies in vivo in mouse, in vitro in human, but we're also seeing targeted guesses that are coming out of a number of good reasons of testing individual genes. Carl's lab tested different checkpoint receptors and found that knocking out CTLA-4 can have benefits in CAR T cells, especially in T cells that are failing from certain patient populations where the cells have shown modes of exhaustion. Others are showing genes that might be less well studied, but have been tested for hypothesis-driven reasons, an epigenetic regulator from a group at St. Jude's. And this is just limited by the white space on this slide. There's now dozens, and maybe even more than that, that are coming out with really good reasons to test, to see which one of these gene edits will make a better CAR T cell. Now, what I want to end with is that if we do these trials one at a time across dozens to maybe hundreds of different genetic edits that are all promising, it'll take a century. It'll take millions and millions and millions of dollars in a century if we did these sequentially. And I think that there's an opportunity to take advantage of the technology, not only to identify the candidates for which genes we want to modify, but to actually take advantage of this flexible technology to test things in pools and measure the effects of individual gene modifications within a pool inside of a patient. So I think there's room for discussion about what the right parameters of size are, but the technology right now allows us to take candidates, dozens of candidates, maybe even more, that are nominated by the literature, nominated by these CRISPR studies in a number of model organisms, and now to go to the point where we actually see their effect in humans. Which ones make human T cells better in vivo in the context of an actual human tumor with its suppressive microenvironment. So now we can introduce these genetic modifications as a pool, test their effects, which one of the cells that has an individual genetic modification gets an added fitness benefit in a patient. And I think this is just the beginning. What we were talking about today is the ability to pick a set of genes that we get rid of, but I want to let you know that this kind of pooled approach is coming down the pike, and you're going to see more and more of this. We're going to see not only the ability to knock out sets of genes, a different gene in each cell to make better CAR T cells, but with CRISPR technology and other forms of technology, we'll be able to add a different gene to each cell, even under regulated control, and we'll want to learn which one of those gene additions is most powerful. We'll be able to do increasingly precise edits. As CRISPR gets better and different flavors are coming out, we'll be able to not only get rid of genes, but actually introduce variants of genes into the genome and see which ones of those genetic variants is most powerful. So we're at an incredibly exciting moment, and it also raises new challenges of how can the regulatory environment continue to be safe and responsible, but also incorporate these new sets of possibilities for the benefit of patients. And with that, I want to thank the people in my lab who've done this work, and thank you again for having me.
the audience here, and I don't see anybody going to the microphones. We're right in time. Alex, stay here. Thank you very much, Alex. It's outstanding to hear all of this science. We read about it, but to be explained. Oh, we have a question there. Maybe we'll take the question and then we'll go to the panel discussion.
Would you propose a straw man trial design so we don't have to take a century to do these studies? Sorry. You said, there's so many options becoming available. So do you have in mind a trial design, a straw man, that could test some of these in expedient fashion? What endpoint would you use and who would you select?
Yeah, it should work now, right? As a straw man, I think what I want to leave you with is, in an experimental setting, with human cells in vitro, we tested every gene in the genome. That's 20,000. I don't think anyone in this room is arguing that we should test 20,000 genetic modifications in a patient. But on the other end of the spectrum, I don't think we have to test one at a time either. So can we take a set of a couple of dozen that have been nominated by preclinical studies, where each one is a really good guess based on what's been done either in a mouse or an immunodeficient mouse with human cells or human cells in vitro? None of those are perfect. We won't know which ones actually work until it goes into a human. So rather than take one guess, could we take a couple of dozen guesses where each cell gets a different genetic edit? We infuse those into a patient, and then over time, we see which one of those genetic edits make the CAR T cells or TCR T cells preferentially accumulate either in the blood or inside of a tumor, suggesting that that is giving the cells an advantage, and that we could use that knowledge either to continue to make cells that have some pool of genetic edits, or that we start to narrow it down to the really good ones for future edits. I think what it means for patients is that patients would get to have the chance to have multiple guesses, and hopefully the winners would start to expand inside the body, not only helping the patient with some chance, although it's a phase 1, we don't know, but some opportunity for the whole field to learn what are the right directions for us to go about genetic edits that will be beneficial in human tumors.
Thank you very much, Alex. And I do not see another question from the audience or online. So we're going to have a panel discussion now, and we're all on this together. That's why we wanted to have all of our colleagues, we wanted to have patient advocates who have listened to these outstanding presentations and will react to it and saying, well, this is what we think about this. And our colleagues from the FDA, we work on this. We all know we have the same goals. And we're very pleased that Nicole and Ewan are here. So can you please come up, and Desiree and Tom and Carl, and we'll have each one, well, Alex and Carl have already been introduced, but we'll let the rest introduce yourselves once we're settling.
I need you to sit in chair 3.
Okay.
We're very well organized, so we'll have seating assignments. See, look at it. And I hope all of the microphones are on. I'm going to move my chair because I want to see everyone. So Nicole, can we start with your introduction?
Oh, yes. Hi, I'm Nicole Verdun. I'm the director of the Office of Therapeutic Products, and I'm happy to be here. Thank you.
Thanks, Nicole. Desiree?
Hello, I'm Desiree Walker, patient advocate.
A little bit more about you.
A little bit more than that. I come to this space of being an advocate because of my lived experience being diagnosed with breast cancer twice. And I like to say that, the reality is the second diagnosis was more challenging, and I experienced a great deal going through both treatment times, but as a result of that, saw many gaps and many needs. And so I've come into the advocacy space because I know that at the end of the day, education is important. In addition to education being important, it's also important for individuals to have access as well as it's also important for the individuals who have been voiceless, i.e. not participating in research, to have an opportunity to do so. At the end of the day, health equity is important, and I want to make sure that it's a conversation that is always had by the brilliant minds that we have.
Thanks, Desiree. Tom?
Hi, I'm Tom Whitehead, and the father of Emily, the first CAR-T patient. And since Emily had her success, every day since then we've tried to pay it forward. And from getting calls from patients from all over the world, when Emily's treatment first hit the media, which went international that day, we just tried to help other families have the same success as our family, and again, make sure that these treatments become globally accessible to the patients who need them.
Thanks, Tom. Ewan?
So I'm Ewan Wu. I'm director for the Office of Pharmacology Toxicology. It's one of the sub-offices within the Office of Therapeutic Products at CBER.
Nicole, you wanted to say something before?
No, I appreciate, I just wanted to say that I was with the FDA. For those that are online, I know that there are several. And I'm a pediatric hematologist, oncologist by training as well.
Thank you very much, Nicole. And Tom, you got a little bit emotional talking about your daughter, about being the first one to get the CD19 CAR for a child. And can you talk a little bit more about the decision-making that you and your wife had to go through when you decided that Emily would get this therapy, and how much of the potential to contribute to the science was part of the decision-making?
Yeah, I would like to start off by thanking Dr. Marks and Dr. Scott Gottlieb, who was in charge, and their teams, because it was ready for Emily. The day we were going to be sent home on hospice, to enjoy the days we had left with Emily is what we were told. CHOP had received permission to try their first patient the day before, so it was within 24 hours of the FDA approval and the IRB approval that it was ready. So we're very thankful every day, and thinking of that when I'm here is makes me feel blessed to still have her. But we had done 22 months of standard treatment that failed Emily twice. Some really tough times. I think a lot of you know in the first week of treatment, Emily almost lost both legs to necrotic fasciitis. When she was relapsed 16 months in, it was much harder to hear that than the original diagnosis. And by that time, when they told us to take her home, she didn't look that sick. And we had done the research. We had been to the Children's Hospital of Philadelphia twice for second opinions. We had a chance to do maybe 2 weeks worth of research on what CAR-T was. They had told us that CAR-T, that they were ready to treat, but it wasn't going to be approved in time for Emily to survive long enough. So to hear that it was ready was amazing to us, but we were very hopeful that it would save her. We knew, we were being told by many doctors that patient one in a phase 1 trial don't survive. And whatever we did, we wanted to help her, but we also wanted to be part of helping move the science forward so other families didn't have to experience what we were going through in the future.
And you actually indeed did for many patients. And I want to make one comment on what you said. I think we doctors fail patients if we don't give them the right treatments, we don't know the science. Patients do not fail treatments. So I wanted to go to Desiree. And Desiree, you've gone through 2 different standard of care therapies for breast cancer. And now we take you out of your comfort zone and we ask you to be here in a panel talking about cell therapies. Some of the presentations I had trouble following because the science is so cutting edge. How do you think patients should go around to understand what's happening? You said that we've been here before with other therapies that were complicated at some time and then they became standard.
So yes, I will definitely say that you've brought me out of my comfort zone and element, but I'm always interested in learning because at the end of the day, unfortunately, disease is still in existence at the end of the day. And I would just say that for me, although this is very complex, I do, and I've appreciated, listening to Tom on the experience that I've had, listening to him, acknowledge, the difficult place that, in his case, as a parent, but also an individual. When you hear that you've been diagnosed with something, it's very traumatic. And your first instinct is, am I gonna die? And you want to know that there's something that's going to potentially give you more time with your family, friends, and perhaps even fulfill dreams that you have. So, for me, I think that, this is very complex. There's lots to think about, but I really want to focus on the fact that individuals had some dream before they had a diagnosis. And it is so important for people to feel as if there is still an opportunity for them, and they don't necessarily, and I appreciate, the opportunity that Tom and his family had, given the news that they were given, that there still is an opportunity, and to hear that, his daughter is a freshman now, I can't even imagine, what that is like to know, wow, how things have come. But I think, we can understand what this is, because one of the things that we should do, in science, yes, we want advances, but we should be able to make sure that we're speaking in plain language, so that individuals actually understand, to the extent that they're able, without having the full science background, what it is that they're potentially signing on for. I think that most people will accept. I know in my situation, even though I dealt with standard of care, I knew that there were risks, but I wanted to be able to see my children grow up. I wanted to still be able to do things, and so I took that risk. I'm thankful that I'm still here today, despite the aggressiveness of the cancer I had the second go round. And so I think, what I would leave is that when I have had opportunities to talk with individuals who have had different types of cancer, not just solid tumor, and complex cancers in terms of blood, etc. I'm always telling individuals it's important for you to try, even though it's a very traumatic time, to try to understand what this disease is to the best of your ability, because you want to make informed choices. Especially when we have research, it's important to know, especially when you're at the phase 1, we don't have a lot of answers, but we're trying to get some information. And you want to be able to make an informed decision at the end of the day. And so I think that, thankfully, we have the World Wide Web that offers a lot of information, and we have organizations that focus on the diseases in order to support you, and we have patient advocacy organizations as well. And I just encourage people to tap into as much as they can handle and process as best as they can, so that they can make an informed decision about their treatment options and move forward.
Thank you very much, Desiree. So we've heard about CAR-Ts, we've heard about TCR therapies that have different gene knockouts. Nicole, I wanted to ask you a little bit, going to what Desiree was saying, that we need to be able to convey things well and have the right terms. How are these products considered? That they're cell therapies, they're gene therapies, combinations of either, and do you regulate them differently from other type of therapies?
These are considered cell-based gene therapies. So modification of a cellular genome or expression of a transgene is considered a gene therapy. From the regulatory perspective, as Ewan alluded to, we have a super office structure and have several sub-offices. So from a regulatory perspective, from the CMC standpoint, there might be differences in terms of classification if you're classified as a cell or a gene therapy, in terms of the reviewers that are looking at those applications. But the super office structure is really wonderful in that it really fosters communication across all of those offices. And allows us to have consistency in terms of how we are regulating these things. So from a regulatory perspective, that classification in itself does not lend itself to differences per se. It really is aspects and the characterization of the product itself in terms of from a safety perspective, release testing, and otherwise. And we really have a great structure in place and a wonderful leadership team and reviewers, many of whom are here, that really maintain our ability to regulate these in a consistent way.
And as you can see, we want to keep you busy with all of these advances.
We are up for the challenge.
Yes, yes. We know you are. Ewan, from the farm tox perspective, what are the primary concerns when we're talking about heterogeneity in cellular therapies that's intentionally introduced by design?
So thanks for the question, Tony, and thank you also to the organizers for having us here today. It's been a really great discussion so far. So from a non-clinical perspective, when we're thinking about heterogeneity that's intentionally introduced, I would say that having heterogeneity itself is not necessarily a problem. I think that it's just important that it's done in a controlled way and that it's characterized. And so I'd say the challenge is that sometimes it can be increasingly challenging to characterize the safety as the heterogeneity of that product increases. And I think that's certainly the case when we start talking about including multiple edits at the same time. So for non-clinical, I'd say the first consideration would be understanding the safety of the particular gene targets, right? Many of these gene targets are being selected with the intention of increasing the functionality of the T cells. So that might be to increase the T cell activation, increase proliferation, maybe decrease the exhaustion or increase that persistence. And these are all very desirable features of a T cell product, but I think the flip side of that is these are the same features that can also drive the toxicities. And so you end up walking this fine line between trying to create a more active T cell product, but not driving it to where you'll have overactivation of the T cells, or you may be generating a transformed cell and increasing that risk of secondary malignancy. And so I think this is an area where it's important to very carefully select those targets and have some safety to support it. The other aspect of this would be to understand the safety related to the mechanism by which you may be introducing that heterogeneity. So if we're taking the example that we're talking about today of using gene editing to create these targeted edits in the genome, you're introducing double-stranded breaks, and there are some safety issues that are associated with that, right? There are the risks of off-target editing, of genomic translocations, and some of these concerns related to the genomic integrity of the product usually increase as we're talking about increasing the number of edits or doing multiplex editing. And so these are issues that would need to be addressed. There are a lot of tools now, both on the bioinformatics side and the non-clinical side to start to assess the safety of this. And I think the other part of this is the manufacturing side, very carefully optimizing the manufacturing process to have control over how many edits you're introducing per cell and optimizing those guide RNAs to reduce the risk of off-target. So I think there are some challenges there, but there's certainly tools that are available to help address them.
Ewan, that's very informative. And I'll turn to Alex because it goes back to what Alex was presenting. You presented an example of one gene, RAS-A2, that can have efficacy, and then you say, well, but we have so many of them, it will take years to test all of them one at a time. But then Ewan is telling us, well, we also need to assess safety. So is there any preclinical model where you think we could do the safety testing, or it's that something the same as the efficacy, where the human model is unique and will not be able to recapitulate it?
It's a great question, and I don't think we have a perfect answer. I think as a field, we've accepted that there are certain things we learn from preclinical models. And there's a standard on validation runs that has been used for what's reasonable to move forward into clinical trials. We see some signal, and there's no glaring signs of toxicity in the admittedly imperfect models that we use, which means if we're going to use a human cell in vivo, currently that means going into an immune deficient mouse. And so that there's only so much efficacy and toxicity that we're able to learn there. I guess my fundamental view right now is that as a field, we have solved the problem of genetic manipulation largely. There's still improvements coming, but any genetic modification we can dream of, we can fundamentally make. What we haven't solved is, and I don't know if we'll ever solve, is the problem of how will we test those in a way that tells us something about what's going to work to cure a patient. And I think that no matter what model we build, whether it's a mouse, if we try to make more complex disease models, none of them will ever be a perfect substitute for what we will learn in a clinical trial. And so I think we have to find the right balance of what have we convinced ourselves is a reasonable thing to take forward to a clinical trial, and then actually learn the lessons about which one of those genetic edits that we've made actually brings some benefit to a human patient.
And Carl, continuing on the same theme, so do you think this is, one would take this approach to patients? Well, how would you take this approach to patient to competitive testing of different gene edits? And do you think we would be able to select from within a certain number of patients, we'll be able to select the gene edits that would be more favorable for the cell therapies for everyone else?
Well, thank you, Tony. I mean, technology has really advanced to be able to deconvolute a tumor biopsy and single cell sequence to know, the state of those cells and what genes were modified. So that's a huge breakthrough we didn't have a few years ago. Now we can't do what Alex has been able to do in vitro, which is screen the whole human genome, because what we have to look at in patients is the feasibility of a biopsy where you recover these patients. I mean, once we infuse cells, there's one thing, which is the efficiency of the initial engraftment of the infused product, and then how fast they proliferate. So that's a variable that can be changed by genetic manipulation, and that's one thing that could be improved. But the other then is this issue of, which really is a holy grail, of getting into the tumor and proliferating and killing tumor. Now that, so I think we're going to need to look at the blood pool and then the tumor. Now, the thing is, we can only have a tumor of a certain biopsy size. So that means we can't recover the entire, we're going to be sampling a population. And if it's a needle biopsy or an excisional biopsy, that will define the limits of our power to really screen through a number of genes. So that will be, I think, the fundamental calculation of how many genes to test initially in a patient. But, and as Alex said, I mean, we have several dozens of genes, hundreds actually now, that have by the various groups been shown to work. And unfortunately, I don't think there's going to be any one, because different tumor microenvironments have different ways of turning off T cells. And so what might be ideal in a glioblastoma might not work in a melanoma or be ideal in pancreatic cancer. So I think we're going to have a series of trials, with the sampling powered based on what we can, depending on the tumor size and the biopsy, to be able to actually know that we've, actually tested the gene population that we put in. It has to be representative on the biopsy from what was infused. And then you can figure out what Alex so nicely showed, who won the horse race.
Mm-hmm. So after these discussions from Alex and Carl on the limitations of preclinical models and the need to test in humans, both the safety and the efficacy, I want to go back to Nicole and you and ask about the current challenges that you see from the FDA when you're reviewing preclinical packages for new drug applications.
That's a great question, and I'll take a first stab at trying to answer this. So I would agree that there's certainly limitations in the available non-clinical models that we have to assess the safety of many of these T-cell products. There have been times where they've just not really predicted some of the clinical toxicities that have emerged, and it's something that continues to be a challenge today when we're reviewing the non-clinical data that's submitted for many of these novel products. That being said, I do think that these non-clinical models are an important tool in the product development program. And I think they offer a lot of advantages, especially when we're considering these types of genomic screens where you're trying to identify different gene knockouts that might improve T-cell function or persistence. I think this is a space where the non-clinical models offer really great advantages in terms of being less resource intensive and being able to answer some of these questions more quickly when it comes to anti-tumor activity or cell persistence even. In a very controlled setting, right? It's a very controlled environment when you're working with a murine model as opposed to when you're going into the clinic. And there's also some other tools that are available, like the in vivo imaging to track the cells, being able to sample at very discrete time points post-infusion, being able to do histopathology, things that are just not as easily accessible and more challenging to do in the clinic compared to when you're working in the lab with a mouse model. And so I think this is an important area where, I think the animal models can be useful if you're asking the right questions and if you understand what the limitations are, obviously, of these models. On the safety side, I'm optimistic, when you look at the development of models beyond the traditional immune deficient xenograft model, right? That's probably still the most commonly used model, but it has its limitations given that those mice don't have a fully functioning immune system. But we're now starting to see people use humanized models, syngeneic models, transgenic strains as well. And some of these models have actually been useful to help understand some of the mechanisms underlying the clinical toxicities that have emerged. And so we're starting to see how some of these models can overcome some of the limitations of the xenograft model. So I think it's a really exciting area. I think it'll be great for the field as these models get developed. And I always encourage folks to continue to develop better in vitro as well as in vivo models to assess safety for these products. I think ultimately, I think we just have to understand the limitations of these models, right? There's not going to be a single model that's going to be perfect. They're each going to have their advantages and disadvantages, and you have to carefully think about those when you're selecting the model and think about the questions you're trying to ask. I think the other aspect of it is taking that into consideration when we're interpreting that data. I think that's critically important as well. I think for these products, we're ultimately going to have to accept some level of uncertainty, and I agree with what Alex said earlier that some of these questions about what the safety profile is or what the best edit will be, probably won't be answered until we bring these into the clinic. And so, again, I think it's striking the right balance of doing what we can with the non-clinical tools available to really optimize these products and ensure safety to the extent that we can, and then moving to the clinic when it's the right time.
Nicole, do you want to add to?
Well, I would just say I think that the challenge that we are facing and that we are up to that challenge, it really is a partnership. It's a partnership with all of you. But how can we advance these therapies and get therapies to patients faster while also being able to really track safety and ensure, in ways that we are able to sort of just make sure that we're doing things in an appropriate way where we're not compromising safety. And so we are really up to that challenge and that partnership with everyone here, including academicians, including sponsors, and the FDA. And we have a great team that is really willing to do that. So in addition to what Ewan is saying, I think that really is where we are.
And I'm seeing a virtual question, and I don't know if either one of you want to answer that virtual question, but say, so what do you think would be the optimal number of gene edits? Would be 10-ish? It says, that's the question, what it says, but would be more, maybe that may be more of a question for Alex and Carl, if you don't want to address it. Alex, Carl?
So I think there's one issue, which is, Alex showed you can screen all 20,000 genes. You can make one infused product and put it in 30 different mice that have tumors and be able to biopsy and recover millions of T cells in that tumor, and then you can sequence those, and you can actually then have representation of what the guide pool was in your initial knockouts. We can't do that in humans. Each individual product will be given autologous to the patient, so that it'll be a sampling of one or maybe a few tumor biopsies, but it's not like 30 mice recovering the whole tumor. So that limits, and so we need to actually power calculations of, depends on how many guides there are per gene, and then how many genes are targeting and controls. So that all is, I think, fairly standard statistics now based on the assays that we have that will power and select, guide the design of these experiments.
Anything to add, Alex?
Yeah, no, I think it's just, the only thing, I think that's a perfect answer. I think the only thing I would add is that we might be talking about a few different questions that are layered on top of each other. One is to try to pick, we've done these tests of every gene in the genome to try to narrow down the list. And as we collectively get to dozens, maybe somewhere between 100 and 200 total genes that are looking promising. We want to narrow that down further to try to, in the long run, work towards more manageable and defined products in the future, right? So we're trying to get down to, in some sense, the smallest number. So I just want to be clear that in the first round of these trials that we're talking about, our goal is to set up the experiment so that we're really trying to set it up so that each cell is only getting one genetic edit. So we're not thinking about how many edits per cell. We're thinking about testing a population where each cell on average has one genetic modification. So I think that's the first order. It might turn out that we have to think about some combinations to do further improvements, but that's really beyond what we're thinking about today. We're thinking about how do we prioritize the small number of gene edits that are the most powerful. And it may be that there's some benefit to having a pool of cells where each cell has a different genetic edit, and maybe you don't want to get down to just one winner. Maybe you want to get down to 2 or 3 winners where that pool of cells works collectively well. So I think there's a lot to learn, but the key is that we're just trying to narrow down and test individual gene modifications, but do it competitively so that we can learn the lessons of how they behave inside the same patient and see which ones are really winning out and conferring relative advantages over the other genetic edits.
Thank you very much. And we've gone through the
Well, I also want to acknowledge the patient advocate voice, because we do hear that, we hear, we're willing to accept risk. We're willing to accept risk to advance the field, and I think that's an important perspective. And we do also take those things into account. So I just wanted to add that as well.
And Nicole, that was my point. I wanted to bring it back to the patients, because the reason of all of this is not so we publish another paper, is so we advance the field. But there's one thing that Desiree, we discussed by email and personally, which is these cell therapies are really expensive. And they broaden the healthcare disparities. Patients from underserved populations may have less access to them because you have to have insurance, you have to be in the hospital, you have to have a support system. Some of the things we're talking about are not feasible for many people. But if we make these kind of gene edits, we may be able to make cellular preparations that are much smaller, where we don't need to have billions of cells. We just engineer the possibility of expanding to those cells. And then in the lab, in mice, we have ways where we can get by with not having to do high-dose chemotherapy conditioning or giving interleukin or things like this to expand the cells. All of it would make these cell therapies maybe like receiving a transfusion instead of going through 3/4 of a bone marrow transplant. So, Desiree, how important do you think this aspect of advancing the field for everyone is?
So I think it's very important to advance the field for everyone. I think one of the things, that has been spoken about, is the acceleration. And I want to say that one of the things that is important to recognize time. When an individual hears that they have had a diagnosis, you are now trying to race against the clock. As an institutional review board member, safety is always something that I am thinking of, as I review protocols, et cetera, as well as, but I really think that, we need to think about efficiency, but we also, with the goal that we're going to also potentially advance so that we can have more effective treatments. Because the sad reality is that there are more diseases that are existing today than ever before. And we need answers, especially, for the individuals that, are now being touched by disease. And so, for me, and I think, fellow patients, advocates, families as well, we need to think about cost. It's time. Right? It's, think about the cost, not only the financial piece, which is absorbent, which is going to potentially make some individuals not be able to access, which is very unfortunate. But we also need to think about time in terms of the individual and their families, how that is going to be impacted, because again, with the current system or way of doing things, you have to spend a lot of time getting treated, tested, etc. And when you start to talk about the underserved and the underrepresented groups, they don't necessarily have the jobs or the finances that they can be away for a month, 2 months, etc., as well as to get the support that you need, after. Because it's not just getting to the point where you have the work that's being done, but it's after, because you're now going to be monitored. You have to be monitored closely to see what's happening. So I just think that, you know, it's again, I keep saying it's very complex. I think that, you know, for the patient community, it's going to be important that the work that's being done, yes, we do advance, but let's not leave others behind with the advancements, because it's one thing that if you don't know something is existing, then it's okay, you don't know. But when you know that there are options out there, but you can't access those options, it becomes very difficult. And having a diagnosis, as traumatic as it is, and I just want to also add to the whole conversation about one's mental health, as well as the mental health of their loved ones going through this process. And we just really need to be mindful how we're doing, what we're doing, and think about the end result, not only hopefully having a positive outcome, but the journey through it.
Very well said, Desiree. Thank you very much for all this guidance. And I want to turn to Tom, because the word of urgency, it starts from what you told us about Emily and how you started in this path. And so what's your sense of urgency that we take all of these advances that we're seeing in the laboratory and have the clinical side catch up to them?
Yeah, it's a good question, and I wanted to add on there too. And with Peter's analogy earlier, I feel like as a patient, and the patients that I'm hearing from now since we started the Emily Whitehead Foundation, the patients feel like they're being thrown into the deep end of the pool, and they hear about the science that's going on behind the scenes, and they want to somehow be connected and be able to have these scientists and doctors throw them a lifeline to try this, even if it means to learn for future patients. I hear from many adult patients now, and they're like, if I could do anything to move research further so less kids suffer from this in the future, I'd be happy to sign up. And I think the public needs to become more aware. I think we need more education of what's out there. I still meet people, you know, all the time that never heard of what happened to Emily or what's going on in this industry until an oncology nurse walked into their room and said, standard treatment's not going to work for you. You need to look up the Emily Whitehead Foundation. And that's where a lot of these calls come from. But again, you know, what Emily went through, I think I've heard from many doctors, it's as sick as you can get without passing away. It was very brutal to watch, but it did change the world. And I can tell you, I'm much more worried as a parent of all the chemotherapy that she had than I am of the CAR T cell side effects. And if something bad happened again with the relapse, we'd be looking into immunotherapy before standard treatment ourselves. And as far as when I hear about the price, so when you're doing standard treatment of chemotherapy and everything else, there was regularly being billed $70,000 or $80,000 a month for standard treatment to our Blue Cross, and when Emily was diagnosed, she automatically qualified for Medicare in Pennsylvania. I think a lot of people don't know that that helps too. But there was several $1 million billed in 2012 to our healthcare, where I hear about the Kymriah, which is Emily's medicine, coming out at $475,000, and as a parent who lived it, that seems like a bargain if you've been through standard treatment for 22 months. So, and I would add that if she went to bone marrow transplant, I don't think she would be coming up on 12 years since she spent a night in the hospital. So I think you got to look at the benefit. And it also showed that we need more pediatric research because it can also help in adults. So we continue to advocate for that as well, because I hear from these parents from all over with every kind of diagnosis you can imagine, and they just want their chance to try these new trials when standard treatment's failing.
Thank you very much, Tom. Those are very important remarks. I want to open it to if there's any questions from the audience in person. I see somebody coming to the microphones. Mike?
Michael Notes from the University of Pittsburgh. Terrific presentations, opening up and discussion. I want to go back to the question that was addressed earlier, which is the straw dog. What setting would you first test this therapy? the argument would be that it would be presumably some CAR T-cell therapy, but then it would be somebody who had bad T-cells, perhaps like CLL. But the question is, could you consider this for another cell therapy, like TIL therapy, patients who you already have an enriched T-cell product that has multiple T-cell receptors, presumably, for cells that have failed largely in their ability to control the tumors. Certainly our single biggest unmet clinical need is in the area of solid tumors, and we need some way to correct those T-cells, and this seems like an ideal approach to do for TIL. I don't know if you'd want to comment on that.
Alex?
I think it's a fantastic idea, and the way I see it is the opportunity is to take any type of T-cell therapy that has some signal that it is safe at least, and may have some early signs that there's some efficacy, but that it's not yet curative, and find what are the genetic edits that will enhance it and make it more likely to be curative. And actually, the way that I've been thinking about this, and I think the way that this applies to others in the room who have been thinking about this, is that the benefit of the way that we're designing some of these approaches is that there's an inherent modular component. Right? If we make a new library of CRISPRs that will target these set of, what we're calling dozens to just over 100 gene candidates, that pool of CRISPR guide RNAs could potentially become a standard reagent for clinical trials that then gets used in different centers in collaboration with TILs. So you could take TILs and make a genetic edit in the population. You could do it with CARs, with a CAR that seems like it might be promising but is not yet curative. You could do it with a TCR, and you could do it for different indications. You could do it, as Carl alluded to, for glioblastoma and pancreatic and ovarian and sarcoma. You could try it for a few different things. And we may learn that there are different enhancements that work depending on what the antigen receptor is and what the target organ is.
Tom Whitehead brought up the issue about how difficult this therapy is. And for virtually every T-cell therapy that I'm aware of today, we use non-myeloablative chemotherapy, which is almost certainly the hardest part of the therapy, in some ways the hardest. And the question is, could these strategies allow us to limit the need for this kind of aggressive chemotherapy that might make it more available and accessible for patients?
The question is, if we were able to engineer the cells so we do not need the chemotherapy conditioning, would this be better for patients? For Emily, obviously.
Yeah, I mean, there's a lot of parents even right now that have educated themselves on this that would if they have a standard risk ALL diagnosis, they'd much rather try CAR T right off the bat and avoid the chemotherapy. And again, for our family, if we could, with any of us, if we have a cancer diagnosis and we could try one of these new advanced therapies before we do the standard treatment, I would sign up for sure.
Right. We'll take two other questions.
Jeff Summers, I hope I have a job tomorrow after I ask this question. So I'm going to set the stage here a little bit. And I can't tell you how thrilled I am that my CBER colleagues are up there, and there's some fantastic scientists are up there, and that you are trying to move this field forward, because I just see it as potentially paradigm changing for solid tumors. And I don't know the answer to this. And I am so positive towards the particular CRISPR-Cas screen. Although, Dr. Marson, you said you don't think anybody in here raised their hand with that 20,000 screen unbiased candidates. There was one guy back there who had his hand raised. So there are some diseases that you just don't want to hear that you have. One would be metastatic pancreatic cancer. The other would be recurrent glioblastoma. If I had to give a patient an informed consent document, I would probably be too positive in a CRISPR-Cas screen where there was 20,000 on-target attempts at goal for a T cell that would grow out. And Dr. June, I think, as some of his early TET2 work showed, that patient that was 2 or 3 months down the line that all of a sudden had a response to his CD19 B-cell lymphoma, that TET2 knockout, that was a single clone that grew out, And do you, particularly to my CBER colleagues, and the question goes to all of you, do you think that there are patient populations and indications where you could use a single antigen CAR with 1,000 screens, or 1,000 CRISPR-Cas knockouts or knock-ins, or 5,000? And would there be an acceptable risk-benefit with that kind of an approach? So it was more unbiased and more potential for identifying something like TET2 that you might not really have thought of, even with the in vivo and in vitro work that you've done with the murine studies, et cetera.
Carl?
Well, Jeff, I think it depends on the pool size that you can sample, because, I mean, we don't want to have random readout. I mean, we need to know that we sampled the entire set of genes that were in the guide RNA library, for instance. So, you know, if it's a bone marrow biopsy, we can get 30 million cells maybe. You know, and would that patient allow multiple bone marrow aspirates? I mean, so you could define the scope of what you could screen, for instance, in myeloma or, you know, various leukemias. I think in glioblastoma, really, it's not ethical to biopsy other than the tumor. You know, that's what a neurosurgeon thinks is that, you know, they're not going to have much tissue to analyze. If you do, say, ovarian ascites, now there you might have a liter of cells and then be able to screen a lot of cells. So I think the tumor, you know, this trial design is going to, you know, kind of set what library we can effectively screen. And I think it's an iterative process.
Complete regression and cure. You know, like,
Pardon?
I was hoping along the lines of like complete regression and a cure. I mean, the tumor goes away. Maybe you don't need needle biopsies and sampling if you actually have regression of a metastatic pancreatic cancer.
But, you know, these are going to be phase 1 studies, or it's discovery really, to find out what are promising genes. You know, ethically, we can't tell a patient we're doing this for benefit, because, I mean, we'll have whatever we know if it's a CAR or TCR and if that's had some promise, but otherwise we don't know. It's a safety study and feasibility, really. I think, you know, so I think it's a classic phase 1 design. And we may get an Emily Whitehead response. That would be great. But, you know, most likely we're not, but we're going to learn, and then that'll guide the next design.
I think Crystal Mickle also brought up the idea was, you know, we do experiments all the time. I mean, that's what we do. All phase 1 studies are experiments. Sometimes we don't even have a relevant animal model for toxicology. So it's a first in human, first experiment, start low, go slow. You know, and I think Crystal has brought this idea up. We don't have to give 5 × 10⁶ million cells per kilogram of CAR T, but give a much smaller product and see which one of those clones potentially grows out.
Jeff, those are great comments, and we're being told that take the last question and go for lunch. But I also want to acknowledge that with Jeff Summers, we've been talking about this for close to 2 years, and you've given a lot of guidance to us, and we'll learn a lot from interacting with you. Please.
It was a great panel. Michael Sagailer from NIH. So I have more of a global health policy question for the patients and the regulatory bodies. Are you working closely with regulatory bodies in other parts of the world? Because, you know, these are all coming to regulatory bodies for the first time, and of course that's a challenge. So are there steps along with the science that are being taken to, once these treatments prove themselves, are available places other than the developed world?
Yes, the short answer is yes. We have multiple touch points with regulatory bodies that are outside of the United States, and to ensure that we are all, you know, on the same page and hopefully advancing some of the science at the same time. And so definitely something that's on our radar. Dr. Marks talked a little bit about it this morning, but from a specific perspective on the cell-based gene therapies we're discussing today, yes, we do collaborate a lot. Thank you.
I was just going to add, from a patient advocacy point, I've been able to travel all over the world with Emily, including and talking in front of the EU Parliament, the benefit that our family gained from this trial. And one other thing I'd like to add, when I said that was the sickest Emily got, thankfully Carl and the team came up with a way to treat the side effects, so most of the patients that were treated after Emily don't end up in the intensive care unit.
Well, thank you very much for all these comments. Yeah, just let me close for a moment. I wanted to thank to each one of you, Ewan, Tom, Desiree, Nicole, Alex, and Carl. I think it's been an inspirational panel discussion and presentations. I think it tells us that we all have the same goal of advancing things and bringing better treatments to patients. So thank you very much.
Thank you.
Thank you.
Thank you.
Thanks for what you guys do.
Thank you. Thank you very much to all the speakers and the panelists. You did a beautiful job of explaining a complicated topic, and we thank you for that. After lunch, we'll dive into these issues a little bit more in another session that will be preceded by a presentation from Marcela Maus. And that will begin at 12:50. We did not prepare any sort of lunch program because I know there's a lot to talk about during the break, so we will break until 12:50. There should be lunch buffets right outside for you. Please feel free to bring plates back in here to your seat or use any of the foyer areas. But again, we'll start at 12:50, and thank you very much.
If everyone could be seated, we'll be getting started with the second session.
Okay, everybody can get seated and settled in after lunch. Welcome to session 2. I want to begin I'm Crystal Makel from the Stanford Center for Cancer Cell Therapy. I want to begin by introducing Dr. Marcela Moss. Dr. Moss is the Paula J. O'Keefe Endowed Chair of the Massachusetts General Hospital Cancer Center and Director of the Cellular Immunotherapy Program and Associate Professor of Medicine at Harvard Medical School. She's a hematology-oncology physician and a scientist. After completing her PhD on human T-cell biology under the directorship of Carl June, she has devoted her work to gene-modified T-cell therapies and has conducted pioneering work in multiple myeloma and more recently in glioblastoma, among others. The title of her presentation is Discovering and Developing Next Generation of Cell-Based Gene Therapies, Considerations for First-in-Human Trials. Welcome, Marcela.
Thank you, Crystal, for the kind introduction, and it's a pleasure to be here. I'm so honored that we're all getting together here to talk about how we can make the next generation of CAR T cells work and figure out the best modifications to do so. I'm going to get at some of the questions that were asked in earlier sessions about what would this look like as a straw man clinical trial? What are we really talking about doing this sort of multiple pools of gene edits in T cells? So I'm going to talk about leveraging traceable heterogeneity in CAR T investigational products in human trials. So as you all know, there's significant clinical developmental activity in gene-engineered T cells. I won't go through the slide in detail, but it's just to show you that there is a lot of activity going on, and there are potentially hundreds, if not thousands, of candidates that could make T cell therapies better. Only a fraction of the ideas that are published even in the peer-reviewed literature looking through PubMed will actually make it to clinical trials. So these are on the same axes, and you can see just how much activity there is in the preclinical space supporting potential modifications or edits or enhancements to make T cells better or more functional in multiple diseases. But it would be overwhelming to test all of those in clinical trials, especially if you start to look at, for example, 2 different CARs targeting the same disease group or the same target. It'll be very difficult, if not impossible, to do head-to-head trials to really figure out which one of those confers the most benefit. So I want to go back and talk a little bit about some of the biology behind this. Obviously, you've already heard from Carl June and from Alex Marson, who've done inspirational work in this. But I want to kind of bring it down to what exactly a CRISPR screen is and how powerful a tool it is for understanding genetic advantages under different kinds of selective pressure. So when we talk about an in vitro screen, we may have a pool of cells of interest that get modified with a CRISPR guide RNA library covering any number of genes, anywhere from 3 to all 20,000 in the human genome. And usually there's redundancy where multiple guides are targeting each gene, which really enhances the statistical power that one can derive a conclusion from. One then takes this pool of cells and then applies a selective pressure. That selective pressure can be exposure to a drug. It can be exposure to a metabolic stress. It can be exposure to some physiologic phenomenon. And then after that pressure is applied, you can then sequence the remaining cells and see that the ones that had increased growth or that are present in higher numbers are the ones that had a relative advantage. And that can be tested by sequencing the guide RNAs and quantifying how many there were to begin with and how many there are at the end after the selective pressure was applied. This can also be done in vivo, and here there are more limitations as to how many different genes can be tested because you need to be able to recover those cells and then sequence them in large enough numbers to draw statistical conclusions. And so in vivo, you could imagine you have a pool of cells of interest, apply this guide RNA library, inject them into a mouse or into a small animal model, expose that model to a selective pressure, whether it's the presence of a tumor or a drug, and then sequence after the selective pressure and look at what grew in the screen. And I think the big question here is what is really the complex selective pressure that occurs in a patient with cancer? So when you're doing this in vitro, you can apply one or more selective pressures, but it's usually one at a time, and it's a very controlled setting. In vivo, when we're doing this in immune-deficient mouse models, we don't have the tumor microenvironment. We don't have cells necessarily derived from patients with cancer who've had multiple prior treatments. And so really patients with cancer are much more selective and complex selective pressure than what we can really test in vitro or in vivo. And so an overall strawman sort of schema would be something like this. There's a single antigen specificity in a pool of cells, and then those would get infused into a patient who then would undergo serial blood sampling and potentially a tumor biopsy to then quantify how many cells with each different gene edit are present in the tumor and in the blood over time. And so the idea here is that though each cell has the same specificity, the same binding receptor, they have individual single or maybe two gene knockouts at most to then understand which are the candidate genes that when edited prefer or confer a selective advantage to either trafficking or persistence in the patient. And one of the challenges here is that there are hundreds of candidate genes that could be edited that have preclinical data supporting them that would suggest that they could enhance T-cell therapy. And so the question is, how do we test this? And can we actually test this in parallel, or does it have to be tested sort of all at once? And by in parallel, I mean in different trials in different patient populations, which will be very complicated, or can it be done in intra-patient parallel testing? And so this leads to a potential problem. And so I'm just going to go a little bit historically, and obviously there are much better experts in the room about this, but pharmaceutical development has historically used a reductionist approach. The general idea has been to study a single variable, the single investigational agent, and then draw conclusions on its safety and efficacy based on using the single variable. So this idea is that we can leverage the pharmacokinetics and pharmacodynamics of the agent to establish the relationship between these measurements and the clinical effects that they have. Intentional heterogeneity of the investigational agent has historically been avoided with small molecule drugs because it makes the drug cause and effect associations to their clinical data a lot weaker. And this is particularly true if there's active metabolites. So if a drug has different timelines for metabolites and they're metabolites with different half-lives, it can be difficult to draw conclusions about their cause and effects if they're all being studied at once. Cellular products are a little bit different in that they're always heterogeneous, whether they are gene modified or not. And natural and unavoidable heterogeneity is accepted with blood-derived and cellular products. This is true for things like IVIG, for tumor-infiltrating lymphocyte therapies, and it's also true for gene-modified autologous cell products, which are heterogeneous in terms of patient-to-patient variation of the starting cell product, and also things like vector integration, as you heard from Dr. June earlier. So what would make an intentional heterogeneity approach feasible for gene-modified immune effector cells? I think the first thing is that they're very trackable over time. We have rapid next-generation sequencing that can quickly and accurately quantify specific transgenes, such as guide sequences, for example, or even different CARs. And second is that there's a direct relationship between the quantity of the gene-modified immune effector cell, or GMIEC as I abbreviated here, and their clinical effects. They also have no known metabolites or altered versions, so when you infuse a cell, you don't expect it to have changes in its genome or that would affect it over time. And they can be quantified in any tissue, really, from blood, body fluids, tissues, using quantitative PCR-based techniques, which are extremely sensitive. If the gene-modified immune effector cell is not present, then its clinical effects are not really attributable to them. However, the converse is that if there's low-level presence of a transgene in a tissue, but there's also low-level transgene in the blood, then the causation or the relative causality is much more difficult to prove. So then the next question is, what makes intentional heterogeneity desirable for gene-modified immune effector cells? Well, we already know that there's a lot of biologic heterogeneity in cellular products. However, it occurs somewhat randomly, and this random heterogeneity makes it difficult to interpret the causality. So just as an example, we wrote a single-cell RNA sequencing paper where we sequenced 602,000 PBMC and T-cell infusion products in patients who were responders or non-responders to either T-cell or axi-cell. Just downloading the PDF of the paper that has these data can take a minute on your computer, so I had to screenshot it just to put it on the PowerPoint here. But if you look at the transcriptional profile evolution from the infusion product to day 7 and responders and non-responders, you get a plot that looks like what's in the middle here. We actually called it the Big Bang plot. there's no single gene transcriptional profile or signature that emerges that says, aha, that's what we want to go after. It's so complex. And even in the setting of autologous T cells, where you can use the native TCR as a fingerprint of which cell got which product, they change over time. And so it's very difficult to determine any sort of causality or optimal genetic approach that one would want to take to make the best product using even relatively high numbers of transcriptional profiles and patients who are responders and non-responders. So introducing specific trackable biologic changes into autologous T cell products would really allow the field to make much stronger associations between the underlying biology and their clinical effects. So I just wanted to step back then and take a look at what the current examples of unintentionally heterogeneous cell therapy products do and what their potential side effect profiles are. In the last 10 years, we've learned a tremendous amount about autologous CAR T products, and mainly they seem to have on-target effects. So this is a function of the target antigen and the binding domain of the CAR, and to some extent, the co-stimulatory domain. The FDA has approved CARs that all have relatively similar adverse event profiles in terms of CRS and ICANS. There's some relatively minor differences in the overall scheme of things of how quickly they engraft, how long the side effects may last. But overall, they behave in relatively similar ways. We're not seeing sort of totally unexpected kinds of toxicities emerge with new CAR products targeting the same antigen. The clinical data that we have to date suggests that there's little added risk to an individual patient when testing combinations of CARs that have different signaling domains or that have heterogeneity in their knockouts, including studies such as using 3 knockouts. And I was going to show you some of that data here today just to say we've actually already done a lot of this as a field. It's just slightly different now. And so just to give you some examples, going back more than 10 years, there's been studies of competitive repopulation studies giving patients 2 different CAR T products with different signaling domains. There's also been competitive repopulation studies with the NY-ESO TCR incorporating 3 gene knockouts and some heterogeneity in that. And there's been random gene knockouts that have already occurred with integrating vectors that have demonstrated certain candidate genes that when knocked out seem to confer potential patient benefit and enhancement of the T cell product, including knockout of TET2 and CBER, as you heard about this morning. So this is data from 2011 from the Baylor College of Medicine, who published some of the first competitive repopulation studies. I won't go through it in detail, but the point on the left here is to show you that these were the studies that demonstrated the superiority of including a co-stimulation domain relative to having a first-generation CD3 zeta only CAR. And that was because you could track each different CAR separately using PCR, that way you could identify that the patients who had when they had the responses, they had the engraftment and expansion of the cells bearing the CD28 co-stimulatory domain. The same group then did a second study looking at second-generation CARs relative to third-generation CARs, where second generation included CD28 and third generation was CD28 and 4-1BB. And you can see in these plots whether the patients did or did not have lymphodepleting chemotherapy, that the addition of the 4-1BB co-stimulatory domain enhanced the engraftment of those cells. So this is showing us just some examples where you can have multiple different products in the patient, and you can use these sorts of PK assays to determine which ones are the winner, which ones are the ones that are engrafting the fastest, at which time point over time, and mediating the effects. As you heard this morning also, Carl June talked about the experience with a limited multi-CRISPR editing of TCR T cells in humans. This included knockout of the TCR alpha chain, the TCR beta chain, and PD-1. And you can see in the Venn diagram that the actual product was heterogeneous, where some of the product had all 3 edits and some of it did not. Some of it had single gene edits and some of it didn't. And so this was feasible. The product was heterogeneous. And one of the interesting conclusions is that knockout of the PD-1 actually had a decreased fitness, when really most people in the field thought that blocking or knocking out PD-1 would confer an enhancement. It turned out to be opposite. And so many of the preclinical models did not necessarily predict that this was going to happen. In terms of viral vector integration, Dr. June covered this a little bit, but I just wanted to make a sort of simple point that lentiviral vector integrations in CAR19 can have multiple integrations, and that multiple genes are affected. And so if you group these by complete responders or partial responders with transformed disease or partial responders and non-responders, you can see just by looking at the colors on the right side is that there are many different integrations. And so it can be difficult to interpret exactly how many integrations are ideal and which integrations are most favorable until you have very large numbers of cells, or you have particular patient cases where a clonal event can happen, where you can identify that a particular integration, such as what happened with TET2 or what happened with CBER, that can be what is used to identify a gene. But that's not easy to identify in the product. That is easier, much easier to identify in a patient who has an unusual response or a delayed response to the therapy. So what could the safety implications be from using pools of gene-edited products? I just want to put it out there. What are we really talking about could potentially be safety issues? So I think what's special about this field is that it's not random. It comes down to two specific potential safety issues. The first is that we could potentially make a gene-modified immune effector cell product more potent. And what would that look like? Well, it would look like potentially an increased risk of cytokine release syndrome, or ICANS, which I think many of our clinical investigators and physicians are now quite comfortable managing. If there's an intact native TCR, there could potentially be a risk of autoimmunity because we don't always know we rarely know what the specificity of that TCR is. The kinetics of the expansion may vary by the gene edit, but it's unlikely that more than a month's DLT window would be necessary, at least to identify the early onset toxicities such as CRS or ICANS. That said, we would still expect there to be the 15-year follow-up or lifetime follow-up for potential long-term toxicities. It's also likely, what could be mitigated is that it's also likely that any single gene edit in a T cell is likely to be administered at a very low dose. Because you're using a large pool, you would have a relatively low number of T cells that have a particular gene edit in, say, gene A that could potentially confer this potency. So this would be a way of mitigating the potential risk as part of how this trial would be set up. What's the second risk? Well, the second risk is that there would be a fear that one or more gene edits could lead to a T cell lymphoma. This has been obviously something that's been in the news and on all of our minds recently. But let's go through what the actual data that we know has demonstrated. So previous clinical history with 3 gene edits granted a limited number of patients. No patients have developed T cell lymphoma in that NY-ESO TCR with 3 gene edits. Now, there have been 2 known cases of CAR T lymphoma that were reported with use of a piggyback transposon, and I'll go through some of what that data showed. But there are no reported cases of T cell lymphoma after CAR T with retroviral lentiviral integration, where the CAR integration is a causative or proven accelerant of that T cell lymphoma. And then I just wanted to add, and I wasn't quite sure where to put this comment, but would increasing the number of gene edits in the pool increase the safety risk? And I would say that the number of gene edits per cell may increase the safety risk, but not necessarily the overall number of gene edits in the product, because it's not about the heterogeneity or the amount of heterogeneity in the product. It's about how many gene edits or transfusions or, gene fusions or translocations could occur in any individual cell. And so it's really not about the total size of the library. It's about how many edits are being performed on a per cell basis. So let's talk about the 2 known cases where the CAR actually resulted in a T cell lymphoma. So this was done in Australia, where patients had already undergone a bone marrow transplant, and they sibling HLA matched. An HLA matched sibling was used as the donor for both the stem cell product and the T cell product. And the T cells were collected separately and gene modified using the piggyback transposon system. What was known about these 2 cases is that they had a very high transgene copy number. And so there were on the order of 50 gene edits per cell as these transposons were integrated. And these are the only 2 known cases where the CAR has a direct causal effect on the T cell lymphoma. So this has actually never been observed with either a lenti, retro, or even CRISPR modified T cells. So what is the incidence of secondary T cell malignancies after retroviral retroviral transductions. So this is a paper that was published by the Baylor Group, and it looked like comparing patients who got genetically modified versus non-genetically modified cells relative to the baseline risk of giving chemotherapy, in this case cyclophosphamide. The baseline risk of secondary malignancies after cyclophosphamide is 2 to 5%. The patients who got virus-specific T cells or cells that were retrovirally gene modified was actually exactly the same. And so there was really no signal, at least from this study of more than 300 patients, that there was an increased risk of secondary malignancies after gene modified or non-gene modified T cell therapies. Of course, we all know that this paper came out and an announcement was made in December of 2023 where there were 22 cases of T cell cancers after treatment with CAR T product. Now, 3 of those cases have been looked like it had detected the CAR transgene. This doesn't necessarily mean that the CAR caused the T cell lymphoma. It just means that it was detected, and it could have easily been a passenger. In fact, in the 2 published workups of T cell lymphoma after CAR T cell therapy, one which was in ASH abstracts and one which was a paper in Nature Medicine, which we can go through in detail, it looked like the T cell clone was present in the pheresis product before the CAR T cell was manufactured or infused. So in the abstract presented at ASH, this was a patient who had received prior cilta-cel, and the T cell lymphoma had multiple gene mutations, including 2 loss-of-function TET2 mutations, a PRPRB truncation, and a KP2 duplication, all in the background of a germline variant in JAK3, which is known already to be implicated in T cell lymphoma. So there's not a clear causal relationship to the CAR, either accelerating or causing that T cell lymphoma. In the second case, in Nature Medicine, this was a patient who received CAR-19 therapy, and the T cell lymphoma was discovered in an incidental biopsy of a lymph node that was being resected for lung cancer. And it turned out that the T cell lymphoma had a JAK3 variant, but it had a very low CAR transgene copy number, so it's not clear that the CAR transgene had any effect on the presence of that T cell lymphoma. And that T cell clone also, again, was present in the blood before the CAR T cells were infused. So right now, there are no reported cases of viral vector CAR transgenes causing T cell lymphoma. Regardless, what are some of the potential measures to enhance patient safety that could be done if one wanted to do a gene-edited pool of intentionally heterogeneous CAR T products? And I'm going to divide it into 4 sections and then talk about each one on its own. So first, one can think about the product design. We're talking about autologous T cells with a validated antigen target binding receptor. One could consider knockout of the endogenous T cell receptor. One can consider using an immunogenic CAR or TCR construct such that there would be sort of a built-in rejection that would be expected. And one can consider adding a safety switch. Now, I just want to make the point that also, just because we're talking about all of these doesn't mean that one would have to do all of these in order to proceed. It's not necessarily exhaustive of all the things that can be done, and some of them could be done for characterization or in validation runs and not necessarily in each different product. The second thing is that one could consider how does one design or characterize the product. And so one could design a rationale or justify a rationale for each gene edit. And one can use computational selection of guide RNAs to minimize potency and reduce off-target effects based on these computational tools that have been developed. One could also perform a transformation assay from validation runs done at large scale from, say, healthy donors and test them either in vitro or even in mouse models just to get a sense for what the potential incidence could be of a T cell lymphoma or sort of unrestricted growth of a T cell product could look like. In terms of protocol design, one would of course select the patient selection criteria very carefully to optimize for patients who have potential benefit and minimal necessarily excess risk. One can design dose escalation strategies. One can have things like safety pauses, stopping rules, and patient staggering for follow-up in between. Have external data safety monitoring boards and include safety monitoring and long-term follow-up as is done for most early phase CAR T trials. In terms of the product release testing and manufacturing, one could imagine something having like standard viability, sterility, and dosing, but also potentially limit the vector copy number per transduced cell to minimize the potential for multiple gene edits that were unplanned in any one particular cell. So if we go through them in more detail, in terms of product design, autologous T cells we already know have a very low transformation frequency. If we use validated antigen target binding, that still enables us to go after multiple different CAR receptors and TCR receptors, which already have known safety profiles in humans, to really focus on the primary variable being the heterogeneous pool of gene edits that are done on a per cell basis. One can consider knocking out the endogenous TCR. This would mitigate the risk of enhancing an autoreactive T cell or T cell receptor, and it would allow perhaps for post-manufacturing collection of or selection of successfully gene-edited cells using magnetic separation. However, it does entail an additional genetic modification per cell, so then one is talking about one fixed gene knockout and then also one variable that would come from the library. Using an immunogenic CAR TCR construct has the benefit of potentially limiting the duration of engraftment, particularly in a solid tumor where we have not seen durable engraftment or 17 years of follow-up or 10 years of follow-up. But it would also potentially make it so that it's harder to figure out what the winner is if we're also getting immune rejection after only 1 month. And finally, one can incorporate the use of a safety switch, such as a truncated EGFR or an ICAS-BASE9 or various others that have been proposed in literature. One cautionary note, though, is that there's relatively limited clinical evidence supporting that they would work, especially in the case of a malignant clone. What about characterization of the product and the gene edits? Well, I think we would need to provide comprehensive rationale for gene editing, for each gene edit that is pursued. This is actually potentially not the difficult part to do. The harder part is assembling all of the data from the hundreds of gene candidates that have been proposed in the literature. One could also use a computational selection process for each guide RNA in the library. So typically FDA would require systematic experimental validation, but it's really not practical for guide RNA libraries, particularly as the libraries get larger. However, the computational predictors to maximize potency and minimize off-target effects keep getting better, and some of those could be experimentally validated using select guides. The size of the library should really be considered based on feasibility of the post-infusion analysis. So this is something that Dr. June was getting at, is why not do all 20,000 genes with multiple guides per gene? It really becomes about how many cells can you collect back from the patient and get a signal rather than just sort of random, because each patient basically has to be their own screen. You can't actually do pooling of those patients' outcomes the way you can with small animal models. And I would also posit that one could consider, in order to enhance the safety, avoiding intentional deletion of genes involved in DNA repair, including genes like P53 or mismatch repair genes, such that there wouldn't be a sort of snowballing effect of different gene edits or mutations that could occur in the product. How could we approach manufacturing and release testing of the investigational product? Well, I would presume that one would adhere to standard viability and sterility criteria. And then we can measure vector copy numbers as copies per transduced cell, and they can be measured and they can have upper limits. And they don't necessarily have to be the same for what the CAR or antigen receptor is, so we usually have established copy number limits for those. The library could have a tighter restriction such that we would exclude samples that had many, many gene edits per cell. So for example, if you had only 2 or 5 or 10 maximum multiplicity of infection or vector copy number of the library vector, you could set a limit such that there would be very few cells, if any, that would have more than the intended 2 or 3 gene edits per cell. In validation runs, one could also perform a transformation assay, such as measuring cytokine-independent growth. This could be done with a sample of the final product from validation runs to demonstrate time-limited independent growth over several weeks. This could be done in vitro and in vivo, and it could be done with higher MOIs or copy numbers than one would intend to put into a human trial to see what the upper limits of those could be. It is impractical to do this as a release criterion for autologous products only because there is a patient waiting, and if one has to wait those many weeks for either an animal product or an in vitro, then the kinds of patients that would be eligible for a phase 1 trial may never get their product. How do we think about dosing when it's a multi-in-one product? Well, we really think about dosing so far as the number of CAR-positive cells, and this is true when it's unintentional heterogeneity, and I would expect it to be true also when it's intentional heterogeneity. And this would not really change if there's different integrations or different gene knockouts in the overall infusion product, because each cell that has an active transgene of the CAR or the TCR would be a potential dose and active agent. What about protocol design? Well, patient selection criteria for phase 1 studies is typically patients with advanced cancer who are heavily pretreated. One can think about focusing on patients with solid cancers because they're less likely to have had multiple chemotherapies that affect their hematopoietic stem cell compartment, potentially less things like CHIP or underlying mutations in their T cells. One can consider excluding patients with hematologic malignancies just because of their higher risk, and even potential higher risk of downstream T cell malignancies, which we see can occur. In terms of dose escalation, we could think about the proportion of T cells containing an additional gene edit would begin at a minimal level, and they could increase as in subsequent cohorts as initial safety is established. One could have pausing and stopping rules based on both DLT, and of course occurrence of a T cell lymphoma would trigger a much deeper mechanistic review and pause until that is identified as a mitigatable strategy. Obviously, having independent data safety monitoring and long-term monitoring could also be included. And of course, evaluation of the individual T cell subpools to associate specific gene edits with any significant safety events would be part of the goal of the study. So just in the last 2 slides to show you an example of what this could look like, here's an example of a library vector construct. So in this case, there's a library guide RNA in one position. The guide library could include guides targeting cytokines, epigenetic regulators, metabolic regulators, signaling molecules, surface receptors, transcription factors, and potentially intergenic controls to understand what the signal-to-noise ratio is in the system. One could also include a fixed guide RNA, such as knocking out the TCR, and one could include a suicide gene and a marker gene to assess for both transduction efficiency and gene editing, and potentially include a suicide gene, as I mentioned. So the concept would be something like a first-in-human phase 0 or phase 1 trial to really discover the drug. These would be patients with relapsed refractory cancers getting a single dose of autologous T cells transduced with a single antigen receptor with a known target specificity and a library of secondary modifications. Endpoints would, of course, be safety and manufacturing feasibility. Secondary endpoints could be clinical responses, and then the scientific or correlatives would be to identify which gene signatures and knockouts are associated with persistence in blood, infiltration into tumor, overall response rates, and toxicities. These are just examples. They're not written in stone, but it's just examples of how we're thinking about it. And then top hits and signatures may also differ across indications that could be incorporated for further drug development individually. So again, this could be a modular where the same vector library could be used with different kinds of CAR or TCR transgenes to identify different effects across different kinds of tumors. Oh. I can't see the last slide. Okay. So just in summary, intentionally heterogeneous pools of autologous T cells appears complex, but the safety concerns are largely limited to the on-target effects and the T cell editing. And the potential safety risks is driven really by the number and type of gene edit per cell rather than the heterogeneity of the millions of cells in the infusion product. An intentional heterogeneity really offers the same or higher potential benefit to an individual subject participating in a first-in-human clinical trial as a typical CAR T cell study does that has unintentional heterogeneity. I'd like to close. Thank you.
Okay, I think we're going to hold with questions until after the panel. So I want to invite the members of the panel up. Nicely done.
We have side seats somewhere, so
yeah.
Hi, Asha. Thanks for being here. All right, so I'm very excited to welcome our panelists today. Let's just go down the row and have folks introduce themselves to start out.
Okay.
Rick?
My name is Rick Bangs. I'm a survivor of muscle-invasive bladder cancer. For those of you who are not aware, that would mean that I've had my bladder, my prostate if I'm a man, or I'd have a complete hysterectomy if I'm a woman, and 1 to 2 feet of my small intestine have been removed. So my revenge on the snake that bit me is my advocacy work. And I have done advocacy work starting with SWOG in 2009. SWOG is funded by the National Cancer Institute. I've also done work for the NCI itself, for the NCCN, the Bladder Cancer Advocacy Network, and a number of other organizations.
All right, Asha.
My name's Asha Das. I work at the US FDA at CBER in the Office of Therapeutic Products, Office of Clinical Evaluation, and I'm the Division Director for Oncology, and a pleasure to be here.
Thank you. Phil.
Yeah, Phil Greenberg. I guess you could say I'm a survivor of the NIH funding gauntlet. But I'm the head of the immunology program at the Frederick Cancer Center. I've been involved in cell therapy and genetic manipulation for decades. I'm older than I look, I think, actually. But it's been a wonderful journey, and it's gotten really remarkably exciting now.
Hi, everyone. My name is Anna Quillis. So I'm the branch chief for Gene Therapy Branch 4 in the Division of Gene Therapy 2, in the Office of Gene Therapy, in the Office of Therapeutic Products at CBER at FDA. And just for everyone who doesn't know, of course, we went through a reorganization recently. So Division 2, so Gene Therapy Division 2 is the division that reviews the ex vivo modified cellular gene therapies.
Great, Marcela. So Chris.
Hi, I'm Christopher White, mucosal melanoma survivor, thriver, and advocate, diagnosed in July 2018. Spent 18 months doing every therapy that existed before finally getting into a clinical trial for TIL therapy, and I was the very last patient to be dosed in the now FDA-approved Amtagvi. And I ended up having a brain metastasis right prior to the trial, which I was able to stabilize and get back in, but I was one of the 12 mucosal melanoma patients as well as one of the 6 responders, and within less than a year I had a CMR, complete metabolic response. But I'm very proud to be the very last person to be dosed in that trial, and I've since dedicated my life to being an advocate for cell and gene therapies, advanced cell therapies, clinical trials, self-published author, and speaker.
Thank you, everyone, for being here. Distinguished panel. So I thought we could start with Phil, who we didn't get a chance to hear from today, but he certainly has every bit of the deep expertise as our speakers that we heard from today. And just to maybe step back a bit and hear your thoughts, Phil, on, we've seen so much clinical progress in the last decade or so, but arguably there's been even more progress preclinically that maybe the public is less aware of. Can you just give us an idea in your mind of the possibilities and the vast universe of enhancements available?
Yeah, I think what we've talked about and what Alex is alluding to, this has really been a sliver of the scope of what really can be done. And clearly the most simplistic strategy is to knock out a gene and ask, when you eliminate that gene, what happens to the cell. But preclinically, of course, all of us, Alex as well, have all been looking at other strategies which are much more sophisticated and which are just waiting for the opportunity now to get in line to bring forward to the clinic. And I just to give you some sense of that, obviously, rather than knock out genes, you can turn genes on and you can regulate their expression by doing base editing upstream. And that's the kind of strategy that will unquestionably be following this kind of knockout strategy. But there's a whole field of synthetic biology that all of us are really actively engaged in, which really changes the way a cell sees its environment. And that's really important. So I think the simplest thing, people have talked about PD-1, it's an off signal. People have tried, as you heard, to knock it out, to eliminate it, to get rid of that off signal. But what you can do is actually take advantage of the fact that it gets a signal, and you can change the way it's wired so that instead of turning a cell off, it actually turns a cell on. So the PD-1 continues to see its ligand, but it provides an activation signal. We've done the same thing with, for example, a death signal. T cells, as we've talked about, commonly disappear. Typically, that's mediated through a receptor on the cell surface. And we've changed that into a signal that makes the cell more viable, more metabolically fit, able to proliferate. And you can change all of this wiring, and that really will change the way cells work. And the question is, we and others have now shown so many of these that really can change the biology of a T cell in a tumor. The question is, which one do you take forward? And the problem is having to answer that by, if it's one, I think then we're back to something that Alex used, it's a guess. And that's not good enough. And I think this opportunity to essentially have a race in where you put cells against each other, I think really will change the outcome and help us understand in what setting which one of these strategies will really work. And I do want to just say that one last thing is that I was really taken by something that Desiree said earlier, is that, well, we consider this a race between the cells. As Desiree said, for patients right now, they're in a different race once you get a diagnosis of cancer. And we appreciate that, and that's really the reason for wanting to push all this forward and to move it, to change the pace at which we can ask and answer questions, because that's a race that we want to make sure people can win.
Yeah, absolutely. And, I think the premise, the basis upon which we're proposing this is that we learn more quickly.
Absolutely.
But it is possible that we could also benefit patients more quickly too. We just don't know because we haven't done it. So Phil, to follow up this vast universe of possibilities, and then you say, if we had to pick one, it would be a guess. Why aren't our preclinical models, how good or how bad or how limited are we at being able to pick the winners using preclinical models?
Yeah, so I spent several decades working only in mouse models, and they're enormously informative, and what they tell us is, does this provide a signal? Does it change the biology? And we can get a signal that it works, and we can get a signal of activity. What we can't really predict from any of these models is, will it be efficacious in people? We know that it'll hit its target in a person. We can't really interpret what will work, and that in part reflects the different biology of many components of the immune system in the mouse versus man. It also reflects the fact that the tumor microenvironments are different. In mice, when we look at tumors, these are tumors that have typically, if we're not over exuberant, they may be a month old, but that's about it.
Yeah.
We're not talking about things that develop over years. And so there's a whole different biology in human cancer that you really can't model in any of these systems. For example, the human tumors put into mice, those are there for typically 2 weeks. So you're not talking about systems that are really analogous, but they do tell you that you have something that has a signal, that has activity, and that's what you want to take forward and understand. And we get some evidence of safety, but not adequate evidence of safety. So I think, as Marcela talked about, there are things that we are doing to try to maximize safety, but unfortunately, much of this is going to have to be answered in people.
Okay, thank you. And you're hearing that from a mouse guy, so. All right, let's move on to Anna. So, given all of the innumerable features of heterogeneity that are affecting the potency of the products we give today, how does the FDA think about these heterogeneous products, and how would you begin to think about regulating something that's that heterogeneous?
Sure, Crystal, thanks so much. So as Marcela also mentioned earlier in her talk, there are a number of different types of heterogeneity that we look at. So it's kind of the first thing that we start to look at. So when we're talking about intentional heterogeneity, where is it coming from? So the first thing we can start thinking about is, say, a TIL therapy. So that, of course, is going to be a very heterogeneous population of cells. So from that perspective, we start to look at the fact that that's coming from the patient. So that is already heterogeneous in the patient. Those cells have already gone through the thymus, they've been educated, so there's somewhat of a certain safety perspective in that alone. Then the next thing you could move on to is, say, the lentiviral vector example of there's going to be a heterogeneous population of cells that have different types of insertion and a different insertion profile. So then what we start to do is we start to go back and look at the vector design for that particular vector and how to compare that vector design to other vectors that have been used clinically. And of course, you know, as Dr. Carl-June mentioned earlier, we have decades and decades of data supporting the type of insertion profiles that we see, what the potential outcomes of those could be, what particular things we could look for to ensure the safety if we have concerns. But even prior to that, you know, we have tons of and even longer data supporting the insertion profiles in HIV patients. Again, to look at what the outcomes of those insertions are. So again, it's a breadth of data that we have to support kind of a safety perspective outside of what's being proposed in an initial product. And then, of course, we have, you know, kind of what's being proposed now, which is, again, a different type of heterogeneity. But we still look at it from the same perspective. And so that is, what available data do we have to support the safety of the heterogeneity that's currently being introduced? And so we rely really heavily on the preclinical studies. And so I should say I'm speaking from a CMC perspective, so I'm a CMC girl. And so, you know, what have we learned from the preclinical studies to support the safety of, say, for example, individual edits? But then also, what type of safety has been introduced during, say, the product design and the manufacturing process, in addition to the testing that's being done? We like to think about the testing as more of an insurance at the very end, but we want the safety to be introduced throughout the manufacturing process and throughout the design of the product. And so we start to look at those types of things. There's multiple ways to manufacture products. There will be multiple ways to manufacture the type of product that we talked about today. You know, when we're talking about you know, vector pools, there's multiple ways to use those vector pools to transduce cells that could make it more easily identifiable, the types and the frequency of the pools that are present in this multi-product or multi-attribute product that we're talking about. And so we look at those things, you know, what is being proposed from a manufacturing standpoint, and how could additional safety be built into that. And I think, you know, in addition to all of those things, we really look at how are we confirming the identity of the product. And so from a product aspect, we really want to be able to understand what is present in the product, not only from a control aspect, but also from a patient information aspect. We want patients to understand what is actually being administered to them. And so we're really keen on really, to the extent possible, being able to identify every component that's going into the product that's being administered to the patient, from a safety aspect as well. But again, it's an informed way of understanding what's being administered to the patients. And so we kind of collectively look at all of those things whenever we're reviewing products, every type of product that comes in the door, and not just these types of products, but just when we're introducing additional levels of heterogeneity that can increase kind of the number of tests or the number of assays, or even the additional characterization that needs to go into the assessment of those products in order to meet those bars and in order to allow us to understand exactly what's present in the products. Just another piece of that as well, I think it's important for products that have this intentional heterogeneity to understand what's in the product from a perspective of we talked a lot about assessing what's coming out of the patient or what's growing in the patient. And so in order to understand the meaningfulness and the impact of those results, it's even more important to understand what actually went into the patient in the first place. And so that's really what we're looking at as well, because we want these clinical trials, if they go forward, to be able to be meaningful. And to understand, the impact of that is to understand what's going into the patients. And so that's kind of collectively, kind of what we're looking at for the most part.
So the better the investigator can characterize the product at the greatest steps possible, that's going to give the agency more confidence
Absolutely.
In the product and in the value of the protocol.
Absolutely.
Any comments there on that? That's really informative.
Just the point, I think it's really important to recognize we all fully agree about characterizing the product. Understanding the product is a little bit different. And so I just want to make sure we have that distinction that we all intend to fully characterize the product and fully characterize what happens after the product's given, because that's how we can understand what we need to change up front. But we won't understand it until we've actually given it to somebody, and that's part of the difficulty here.
Great.
Can I add something to that?
Yeah, Marcela, sure.
I'm struck by two things. So I of course understand the desire to have control and an understanding of the product, but I also think that there's so much heterogeneity in people. When I think about even the COVID vaccine, members of my family all got the same 100 microgram dose of the same vaccine, and yet we had completely different side effect profiles afterwards. And so I think when we're talking about biology, it might have been 30 micrograms of RNA, but how our bodies processed, presented, responded to that was really quite variable. So I'm not sure that there's always a perfect relationship between what the product is and what happens to the patient. And then the second part of it is when we're talking about cell therapies, and we've learned some from the random integrations, from transcriptional profiling, is that those weren't necessarily detected in the infusion product. And so I'm not sure that we could have predicted that TET2, that single cell that went into the patient, 10 with CLL, who responded 10 months after the therapy had been given and had this TET2 knockout clone or hypomorphic mutation come out. I don't know that there's any amount of characterization of the product that could have been done to predict that was going to happen. Sure. And so I'm just thinking about, I completely understand, we need to know what we're putting in, but the truth is also it's a little bit like the Harry Potter line, we make a plan, we go in there, and then all hell breaks loose. That there's a little bit of an assumption that the exact way you make the product is going to predict what's going to happen in the person. And I think that's an assumption that I'd like to just consider.
Yeah, I'll just comment that I think it is exactly the randomness that we all know that gives the CMC reviewers at the FDA headaches for autologous products, because you can treat 99 patients and 99 patients do fine, and the 100th patient develops a lethal, ICANS or something. Mm-hmm. So it is exactly that randomness that kind of bodes caution as the reviewers are looking to be the gatekeepers of what goes forward. So it does behoove us to do everything we possibly can to characterize what we can. Maybe that's one way of looking at it, because there is so much that we can't control. Is that?
Yeah, exactly. To Marcela's first point, what if everyone hadn't gotten 30 micrograms, and we didn't know what they got? Say one person got, 5 micrograms of this and 10 micrograms of that, but another person got 15 micrograms of this and 20 micrograms of that, then that would make the data that comes out of that clinical trial even more confounded and more difficult to interpret. And so I think from that standpoint, that's why it's at least knowing what the input is allows us to be able to interpret the output a little bit better.
Gives us a chance. Mm-hmm. Thank you, Anna, and thank you for those helpful comments, guys. Asha. Okay, so Marcela presented a pretty complete menu of different safety measures that could be implemented into a trial testing a heterogeneous pool. From your perspective as a clinical reviewer, what advice would you give to investigators as they're thinking about trying to, use intentional heterogeneity to advance the field?
I think Marcela, you did a great job explaining the different safety measures. I think what I'll do is just start, just explain how we work together as a team at FDA. So we work very closely in collaboration with our chemistry, manufacturing, controls colleagues, and also pharmacology, toxicology. We want to really understand the product. As regulators, it's our job to develop these as safely as we can and not expose patients to undue risk. So beginning with that, we'll of course look at the patient population. Many of them have received prior FDA-approved therapies. And when they come into these trials, we want to make sure that they're well enough to receive the investigational product. If they're receiving toxic lymphodepleting therapy, that they're well enough to receive that as well. So that's going to make the pool a little bit smaller. Then, of course, if it's a TCR, they have to express the biomarker, plus they may be HLA restricted as well. So again, a little bit smaller. Tom Whitehead made the comment about pediatric development, and of course we've got regulations because we want to develop these as appropriately in children as well. So we spend quite a bit of time on that, and that's part of the clinical development plan. And then we want to make sure before the patient gets lymphodepleting therapy that they have the product. So that's also, we make sure that that's included in the protocol. Seems like a obvious step, but sometimes it's missing. And then as Marcela mentioned, we will of course look at staggering enrollment so that if there are any acute toxicities, we understand that. Stopping rules where the trial is put, not terminated, but just put on a temporary pause so that we can understand what's going on and see if any revisions need to be made to the protocol. Of course, long-term follow-up to look for any delayed toxicities and any secondary malignancies. So that's also important. I want to talk a little bit about dose because I think, as Marcela pointed out, and Anna as well, we need to pay a little attention to the dose. Is the dose based on the engineered receptor? Is it going to be based on the edits? What if some cells have just one edit or some have two? So I think we just need to pay a little bit attention to that so we understand. And then we'll look at dose-limiting toxicities. Many of these protocols have dose escalation. So again, we want to understand what's the dose that's being given, and then what's the algorithm so that the toxicities can be managed. And of course, many investigators now used to managing cytokine release syndrome or ICANS. Some of the publications on hemophagocytic lymphohistiocytosis came out last year. And what we see is that, sometimes there may be grade 5 adverse events because HLH was not recognized early enough, or maybe interventions were not instituted early enough. So we pay attention to make sure that's included in the protocol as well.
So that's very helpful. Marcela raised this question of a safety switch, and I think we all think about it in the field, and, it's sort of a reflex that, well, if we're worried, we'll put a safety switch in. But, we all kind of know that they really haven't necessarily been fully vetted. And even if they're vetted for cytokine release syndrome, that might not be a vetting for malignancy. I'm just curious how the panel or maybe the FDA reviewers feel about safety switches.
Do you want me to go first? Please. So yes, exactly, Crystal. That was pretty much exactly what I was thinking, is the fact that a lot of people do think, oh, I'll just put a safety switch in and it'll be okay. But we don't have the data to support how efficient they are. And in particular, again, I'm really going to reiterate everything you just said. We also don't know if it works well for one thing, if it's going to work well for another thing. And in particular, the timing of the administration of, say, for example, the small molecule that binds to the safety switch. A lot of times with the things that are happening, like say CRS or ICANS, they're on different timetables, and so something might be able to work on something that isn't as quick. But if something happens and it's very, very, very quick, that safety switch might not be able to work there. And so, yeah, it's not the be-all, end-all of the answer, is to just put a safety switch in things, unfortunately. I guess in a way we all wish that it would be.
Yeah, and maybe it's an area where our field needs to go back and do some more work to, learn more or make better SynBio for that. So I want to move on to our advocates. So Rick, as a cancer survivor who has navigated many standard treatment options, and now it sounds like has got a degree in oncology. Obviously, you've been able to come to understand a lot of the complexities that is in the space of oncology in general. But you're not, you haven't been around cellular therapies. Probably this is a bit of a crash course for you. I'm curious what you think in your, you've talked to so many advocates and talked to so many patients. How well are patients able to understand the complexity of something like this if it was to be offered to them? And, can they understand it well enough to be able to make an informed decision? What are your thoughts about that?
So when I think of informed decision, I think of shared decision making. So I think that's kind of a foundation and a fundamental. I don't...
Tell us a little bit more about that. I might not know what you mean.
So I see it as a partnership between the clinician and the patient, and I see a lot of calibration necessary. So if Chris and I come to you, and we have the same exact situation, Chris may not want to know anything about the science. Mm-hmm. I, on the other hand, might be a science nerd, and I might need that. But I think there's kind of there's some fundamentals here. As a patient, I think what I need to understand, which I'm still grappling with based on what I'm hearing, is how safe is this going to be? How effective is it going to be? And how durable is it going to be? Those are the three questions I need answered, right? To me, it's like when I buy a smartphone, I don't need to know all the bits and bytes pieces on the software side or the hardware side. I need to know, can I make calls? Can I do emails? So safe, effective, durable, explain that to me. And I understand it's going to be a continuum, and there maybe you can't tell me with certainty, but directionally, that's what I want to know. The actual the way the science works, the biology, while it may be interesting to me as the science nerd, it may not be interesting to somebody else. And I don't know, Chris may be a science nerd too, but to somebody else it may not. And really, at the end of the day, I need to leave understanding safe, effective, durable.
Mm-hmm. Okay. So let me ask you this. Do you get the sense that patients will calibrate themselves with their shared decision making? In other words, for some patients, this risk might be too much for their desire, and for others, they may be more comfortable with risk. And do you think, how do you think that the regulators, is it the job of the regulatory agency to, draw the line at minimal risk, or to allow some risk and let patients decide for themselves what they want?
Yeah, so again, I think there's a continuum, and the thing I always try to, I almost want to reinforce on the clinician side is that cancer is lethal, and so if I don't do anything, I really need to understand what's going to happen if I don't, right? Mm-hmm. And the situations we're talking about are situations in which people are, they have no other options, right? And if there's an imminent result, which may be my death, then for many of us, we may be willing to take the leap, okay? And so I think we should have the opportunity. Now, that being said, there's going to be constructive tension here between, let anything happen no matter what without any evidence, and being really, really rigorous. So we've got to find this balance, but I think it's important to recognize that patients should have choices when they're in situations in which there would be no other option.
So patient selection, in your mind, is a really important part
Absolutely.
of this conversation.
Yeah.
Okay, thank you. Very helpful. Chris, so you faced a dire prognosis before enrolling in a TIL therapy trial that ultimately saved your life. Can you share your personal perspective on balancing the risks of a novel, even potentially unknown therapy against the potential benefits?
Absolutely. You know, by the time I learned about TIL and was, you know, given the option, there was the option of, do I want to live or do I not? You know, because I had filled out my will, and if I didn't do TIL, it was over. And so it's one of those things that if you try, you might make it, but if you don't try, you're definitely not going to make it. And every treatment that I'd done prior, you know, they would always give me here's what, you know, the potential side effects could be. These might happen, they might not happen, but it's not like they all happen necessarily like when you're actually in the clinic getting your infusion. They're usually, you know, once you're home and then you're having to deal with it then and then go back to the hospital, get a hold of your care team and so forth. And all my current, you know, side effects that I had, you know, from my whole entire journey come from everything that I did prior to TIL, both, you know, long-term and short-term. And all the side effects that I actually had from TIL were only for the time that I was in the hospital, you know, for those 12 days. So the lymphodepletion and, you know, mostly the IL-2. But I knew that since I was already in that clinical setting to where I had doctors, nurses, if I needed to do the ICU, I, you know, was there. I mean, I felt completely comfortable with all of that. And so, you know, given a choice, you know, not as a last option, but maybe as like, you know, as it is now as a second option, you know, another tool in the box, you know, then I could look at it of, well, here's the potential side effects, you know, versus, you know, doing this between doing that, and also, you know, the toxicities of it and everything that goes with all of that. So, you know, that being said, I would much rather, you know, be in a setting to where I knew that if I needed help, it was like there, instead of me having to go chase them down. So, you know, from patient perspective, absolutely. I mean, it's, you know, you don't know what you need to know until you need to know it, kind of a thing. And so I guess it's all hindsight. My crystal ball only works so well, you know, but thinking back to it now, I mean, given the option and everything, like I would have totally have taken the risk and just, you know, went full throttle into, you know, a cell therapy from the get-go if I would have known about it.
Yeah. Chris and Rick, are there other things that we should, you know, that you want to share or that we should hear, because your perspectives are so valuable as we think about this?
You know, when it comes down to it, some of these, you know, rare cancers, you know, they don't have a lot of, you know, options, and time is, you know, the most valuable thing that you have, and there's so much of a process to where it's back and forth and insurance approvals and financial toxicity and mental health and all these other things that aren't necessarily considered. One of the things I think about quite a bit is, you know, survival for your follow-up and everything. It's a lot more than just vitals and labs and scans. It's, you know, all the other, you know, effects that go with, you know, along with that. And so quality of life overall, you know, when you're comparing treatments and, you know, how to move forward, you know, with everything. Someone was mentioning to me not long ago that, you know, did you, you know, have a desire to, you know, go back to normalcy? And I was like, normalcy? Like, once you get hit with a diagnosis, you know, that's a terminal diagnosis, like, you know, there's no, everything from that point on is completely different and always will be. I wasn't born like a science nerd and wasn't one growing up, but I've grown to be one over the past few years, and, you know, I'm all about it. So, you know, and I've dove headfirst into all of it because, you know, if you can do an autologous cell therapy and do something once and just, you know, get it over with, I mean, it's kind of like doing the comparison of, you know, do you want to, you have to get in the ocean. Do you want to tiptoe in it, or do you just want to dive in? You know, do you want to slowly rip off the Band-Aid, or you just want to get it off? I mean, like, so, especially with the pricing that they were mentioning earlier, it's, you know, 18 months, over X amount of dollars, you're going to end up paying it, or you can just, or do it one time and pay that lump sum. But if you do a clinical trial first, you have these other approved treatments, that are already there. But if you do all those other approved treatments first, and then you're so heavily pre-treated that you physically can't make it through a clinical trial or something to where, you will have to have a hospital stay and endure like the toxicities, from both, the lymphodepletion and then, but also, the IL-2, the Rigers from that to me was probably the biggest, side effect out of anything throughout the whole process, but it was monitored there. I felt safe in it. They had me covered, a lot of the other treatments that cause, hypothyroidism, my uveitis, cataracts, colitis, all these other things, like I had to navigate those outside of the hospital setting. So if a patient, can physically, I guess, hit all the check marks, because I was extremely, they quizzed me very hard, even though I had passed all my baselines to get back in, just to make sure that like I was physically healthy enough to go through the process.
Right.
And so, if there was a doubt maybe that it was going to be too toxic for me, or things were going to go awry, like they were very sketched out about letting me proceed, but I was.
That's part of the race, is that you stay well enough to be able to try something.
And Chris, this informed consent process, just your perspective, do you feel that you were told about therapy and all the time that it would take? I'm very curious to hear about it.
By the time I went to go sign my consent, I didn't have it in hand ahead of time to be able to go over it and read it, but I wasn't really concerned because I didn't have any options. So as soon as I went to go, sit down with the trial coordinator and go over my consent, I signed it on the spot. And so, my next question was how quickly can I get in for my extraction? And it was a couple of weeks later, but I was wanting to do it like immediately because I was clinically declining so fast that that all these timelines, were not doing me any good. I got wheelchaired into the hospital by the time I went in there. I'd filled out my will. People were coming to visit me like it was the last time they were ever going to see me. So I was ready to do anything and everything I had to. So it didn't matter what the consent said. It could say, Chris, you're going to drink a gallon of gasoline, and I was going to do it. So, but,
that was the race. That was the race.
But that was the risk.
That's what, yeah, that's what we heard. Okay, I think that, does anybody else have any burning comments? Or? Okay, we have one question, virtual question. Are there more efficient ways to test the effects of gene editing other than testing in clinical trials, such as preclinical in vitro or in vivo models, and how can AI/ML bridge preclinical models? So I guess, we talked a little bit about that. Marcela or Phil, want to take that, Phil?
Well, I think, first of all, the answer is yes, and that's all done before we even talk about what we're getting ready to do. So for example, we just did a CRISPR screen looking at knocking out a particular type of gene set. We looked at more than 100 data sets that characterize exhausted T cells out of tumors. We looked at TCGA database. We used AI models to try to analyze that. And then we came out with a set that we then put into mice and actually looked at which one works well, and are now getting ready to try to advance some of this to the clinic. But so all that's already done. So it's not that that's a kind of some parallel strategy. It's a strategy we're all using before we even get to the part that we're talking about here today.
Yeah. Marcela?
I agree with what Phil said. I think one thing that's so unique about this field is that we can actually trace the products. If we use things like gene editing, or if we use particular vector sequence, it's a marker that can be detected with such high fidelity and such sensitivity. It's really a unique opportunity. When I think about other drugs, whether it's vaccines or small molecule drugs for things like depression, There's a black box warning on SSRIs for use in teenagers because of potential increased risk of suicidality. But we don't always know whether the drug is there, whether it has a causative effect. And so we're really relying on statistics and cohorts. And that's because that's the best we can do in that setting. And so it's the only thing we can do. And that's how we wrap our minds around how to interpret this and how to make decisions. What makes this field, and probably the field of gene therapy and gene-modified cell therapy, so unique is that we can actually prove it with a much higher confidence if the vector is there, if the cell is there, and we can measure it, we can quantify it, we can say how many copies per microgram of DNA or per transduced cell in a way that's unique. And so I think some of the systems that we've evolved to think about these things, this presents such an incredible opportunity to really deconvolute and really understand the biology of cause and effect. You perturb A, you get B. And so I think that's just a really unique aspect to this. And I think we're seeing that with the T-cell lymphoma. One of the challenges in understanding the secondary T-cell lymphomas is that the assays to measure the CAR are not widely available. And so even though it's made by the sponsor and it was done in the clinical trials, when it's out there in the community, it's much harder to prove one way or the other because not everyone can send the assay to a clinical lab to get the answer back. But if those things are available, and if we can do this in a clinical trial setting where we are measuring each transduced cell, then it really presents a unique opportunity to understand that biology and make better and safer products.
Yeah, I think it's clear from the conversation today that this field already, but you can imagine without too much imagination in the future that the sophistication that is possible in the field of cellular engineering is so far beyond anything we've been able to do with small molecules and protein therapeutics. It's one of the reasons for the great hope in the field. So this question, I think it's a really good one and steps back a bit. Can the panel comment on where they see the most promising potential breakthroughs coming from towards tackling specifically solid tumors versus increasing T-cell proliferation and other improvements for hematologic malignancies?
Yeah, well, so I think, first of all, this has never been about T-cell proliferation for proliferation's sake. It's about the fact that we know that in a tumor, 2 cells actually can kill a lot more tumor cells than 1 cell can, and 4 do better than 2. So that's really partly why that's being addressed. But the reality is we're all looking at trying to get this to work in solid tumors. And at least in my mind, the real promise is going to be in rewiring cells, that when T cells get into the tumor, they get turned off, and the question is, they're getting lots of cues from their environment, and all of the strategies that you heard about today and the ones you didn't hear about are about how can you change the way the T cell perceives those cues and turns them into strategies that actually enhance activity rather than diminish activity. And that's where I think the real breakthroughs are going to come.
Absolutely. Thank you, Phil. I think, Rick?
So for me, one of the key takeaways here is calibrate. Calibrate the risk, calibrate the regulation, calibrate the communication to the specific context and patient. I really think that's critical here. This calibration of those things is going to help us move forward.
I think that's very, very wise. Absolutely. Thank you. What a great panel, huh? All right, that concludes.
Thank you very much. That was absolutely amazing. I just wanted to, on behalf of UTA, Friends of Cancer Research, and the Parker Institute, just thank all of our panelists, our presenters, most importantly, our patient advocates that were here to really talk about this amazing and really critical topic that we dove into today. And it's one know if we have slides. But it's one that I definitely want to just kind of summarize a little bit, some really critical and important points for take-home messages. And I think one of the biggest areas I think that we want to cover is the need that's out there. You know, there's just such an important need to really begin to tackle issues associated with solid tumors, issues associated with heme malignancies. We heard about the complexity of the cell therapy products, but just as importantly, I think, the opportunity that's presented by these products to really change patient care. And this is really driven by the scientists, by the physicians, the trialists that are here in this room, that are out there and supported by this incredible network of people. And so this opportunity that's in front of us right now can really only happen with the incredible regulatory agility that we see in our colleagues at the FDA. And this is just something that we've seen historically. As science has advances, we've seen the regulatory agility that's been demonstrated by the FDA leading the way globally, and this is something that we're really depending on and really trying to leverage here today. It's an important point, and it really gets to the key and core questions that we found came out today, which is really diving deep into product characterization, taking a look at what we can, there we go, some key takeaways. Just some product characterization associated with the limitations of our understanding of what the important changes are in those cell therapy products, but just as importantly, some of the associated improvements that are coming out of these cell therapy products, the depth at which we can characterize those and move those forward, and then the safety aspects that we can put in front of them. So all of these are absolutely essential in bringing these forward, but what we want to do is never forget the need that's out there in the fields, and it's incredible today to have such an illustrious group of people considering all of these different points and pushing it forward.
Okay. Yes, and I like to bring this back now on the 30,000 foot level and feet level. And this workshop, we think about, brought together all cell therapy expert regulators, patient advocates, and nonprofit organization. And you were all witness to that we could establish over this time and at the workshop a very productive dialogue around the promise and also the challenges of CAR T-cell therapy in solid tumors. Together we explored cutting-edge therapeutic concepts with the goal to unlock the challenges of these complex cell-based gene therapies. And in preparation for the workshop, and you witnessed today, that we all gained a better understanding of testing heterogeneous pools of engineered T cells, their manufacture, and impact on the design of clinical trials. I think we can all agree on that we must continue this work together and ultimately translate what we learn collectively into improved outcomes for patients. And I want to thank you on behalf of Friends of Cancer Research and the Parker Institute of Cancer Immunotherapy. We want to thank the Principal Deputy Commissioner of the FDA, Namandjé Bumpus, and the Director of the Center for Biologics Evaluation and Research, CBER, Peter Marks, for being with us today, opening up our workshop. And I want to mention first the patient advocates. Thank you for participating, getting us your perspective, which is core and center to all of us. Thank you, Desiree Walker, Tom Whitehead, Rick Banks, and Chris White. And we thank the CBER team, Nicole Verdun, Evan Vu, Asha Das, and Anna Quilas. And then I want to thank really, it comes from the heart, our cell therapy experts who are all part of the extended PICE network, Crystal Mackel, Carl June, Tony Ribas, Alex Marson, Phil Greenberg, and Marcela Maus. Thank you very much, everybody. And we thank you for your commitment and the contributions to this workshop. In closing, we all realize that this workshop probably marks the beginning of our journey together as we go forward. The continued dialogue and commitment from all stakeholders is key. We will all need all of you, all of us, to achieve progress for patients. Thank you.